I'm sorry, I was speaking mute.
Hi.
Hi, how are you?
Hi, fine. And you?
Fine as well. Perfect. I will make you co-host so you can share your screen.
Perfect.
Welcome, Dr. Blor. I think that right now, the first one that's going to present it's Dr. Acosta, and then you, Dr. Blor. Am I right?
Okay.
Perfect. Dr. Acosta, can you hear us?
Sí.
Perfecto. I will make you also co-host so you can share your screen.
Perfecto, me dices cuándo.
Perfecto. Listo. ¿Podemos practicar un momento la presentación para ver que todo esté funcionando? Perfecto.
Me dices.
Sí, está perfecto.
Listo, mil gracias.
No, señora, gracias a ti. Bueno, bienvenidos a esta segunda parte. I will begin the presentation in English and then switch to Spanish. Welcome back. We hope you found your lunch very enriching. Next, we have Dr. Juliana Acosta Uribe presenting Genetics in Dementia in Latin America, Lessons Learned from RedLat. Dr. Juliana Acosta Uribe is a research scientist in the Department of Molecular, Cellular, and Development Biology at the University of California, Santa Barbara. She earned her medical degree from the University of Antioquia and later completed the MA and PhD at the UCSB, focused on population genetics and neurodegenerative diseases. Her research investigated how historical demographics processes in Colombia influence genetic risk for hereditary dementias, particularly autosomal dominant Alzheimer's disease. Dr. Acosta, the floor is yours.
Mil gracias. Bueno, espero que todos se encuentren bien. De nuevo, me siento muy feliz de estar presentando en mi país, así sea de forma virtual, y muy agradecida continuamente con el equipo de la Universidad Javeriana. El día de hoy quiero presentarles un trabajo que ha llevado múltiples años de cómo hemos entendido la diversidad genética en el campo de las demencias en América Latina. Antes de comenzar, siempre me gusta hacer unas definiciones importantes. Es importante que nosotros sepamos que cuando utilizamos los términos de ascendencia o ancestría, estos refieren a un término estadístico que es la forma en la que determinamos los genes y las variantes genéticas que un individuo o una población ha heredado de antepasados de poblaciones que llamamos ancestrales, población europea, población asiática, africana, amerindia, etc. Los conceptos de raza, etnia, etnicidad son construcciones sociales que nos agrupan a los individuos según características físicas, culturales, geográficas, que son percibidas como cohesivas. La asignación de la ancestría genética no cuestiona ni debe estar en contra de la forma en la que un individuo se autopercibe como perteneciente a una raza o a una etnia. El día de hoy voy a dividir mi charla más o menos en 3 fases. Voy a hacer una pequeña introducción a lo que es la genética de la demencia, qué tanto se hereda la demencia, cómo hemos hecho el abordaje de la demencia genética en pacientes latinoamericanos, inicialmente en Colombia, luego a nivel de Iberoamérica, y si nos queda un poquito de tiempo, me encantaría hablar un poco de factores de riesgo genéticos para la demencia en pacientes mestizos. Como bien saben, demencia es un término sombrilla, es un término que me abarca un conjunto de síntomas, una colección de diferentes enfermedades donde el paciente pierde una capacidad cognitiva que había adquirido previamente. Para la charla de hoy me voy a enfocar en la enfermedad de Alzheimer, la demencia frontotemporal, pero no podemos olvidar que hay otra serie de patologías que nos causan síndromes demenciales. Demencia vascular, demencia por cuerpos de Lewy, demencia por Parkinson y muchas otras. Me quiero enfocar en la enfermedad de Alzheimer, la demencia frontotemporal, comenzando con qué sabemos de la genética de estas enfermedades. Y lo que sabemos está basado en el paradigma eurocéntrico del conocimiento. ¿Por qué? Porque si vemos los participantes en los estudios genéticos, sobre todo los estudios de descubrimiento genético, estudios del genoma completo, vamos a ver que la mayoría de ellos son de ascendencia europea. Entonces, esto nos lleva a figuras como estas, donde nosotros inferimos que los genes que más nos van a explicar la demencia frontotemporal de tipo autosómica dominante, perfecto, vamos a tener MAPT, progranulina, C9orf, y por ahí hay otros genes raros, variantes que no vale ni la pena mencionarlo. Cuando partimos de la demencia frontotemporal y hablamos de demencia tipo Alzheimer, ya vamos a ver un paradigma europeo, digamos, un poquito más amplio, donde tenemos unos genes mendelianos, preselinina 1, proteína precursora del amiloide, y luego tenemos una serie de genes que nos causan riesgo genético. Este riesgo, calculado por odds ratio, tenemos ApoE, TREM2, como los genes que son relativamente comunes y que nos confieren el mayor riesgo genético. Todos estos genes y variantes los voy a retomar un poquito más tarde, hablando de cómo al identificar o trabajar con otras poblaciones, este paradigma genético nos cambia y el tamaño de estos efectos nos van a cambiar. Pero empecemos en el principio, en lo que nos lleva a nosotros, como profesionales del área de la salud, científicos, a pensar en la heredabilidad de las demencias. Y es cuando nuestro paciente llega con sus hijos y el hijo, viendo al papá así de enfermo, dice, doctor, a mí también me va a dar esta enfermedad, a mí también me va a dar Alzheimer, yo estoy preocupado porque mi papá, mi abuela tuvieron demencia y yo quiero saber si a mí también me va a dar eso. Entonces es el primer momento donde el profesional se ve enfrentado a ¿qué le digo a mi paciente? ¿Será que mi paciente también va a tener la enfermedad de su padre o su madre? Entonces nos toca pensar un poquito, ok, ¿qué pasa si es un paciente con enfermedad de Alzheimer? ¿Qué tanto de la enfermedad de Alzheimer es hereditario? Vamos a ver que en la mayoría de los casos tenemos una enfermedad de Alzheimer poligénica, donde tenemos factores genéticos que nos explican más o menos 2/3 del riesgo que tiene un individuo para la enfermedad de Alzheimer, mientras que 1/3 es factores de riesgo ambientales o modificables. Ese riesgo genético va a estar comprendido por múltiples genes. Vamos a tener unas familias donde ApoE4 nos explica la enfermedad de Alzheimer, aunque pareciera tener un patrón autosómico dominante, seguimos considerando ApoE4 como un factor de riesgo. Tenemos otros genes, como TREM2, que ya lo había mencionado, SOR1, que nos aumentan el riesgo de tener enfermedad de Alzheimer, pero no van a ser causales. La enfermedad de Alzheimer mendeliana, determinada por los 3 genes causales de preselinina 1, preselinina 2 y proteína precursora del amiloide, es supremamente infrecuente. Menos del 1% de los casos son enfermedad de Alzheimer netamente genética. Entonces, en la mayoría de los casos que tenemos enfermedad de Alzheimer, sobre todo cuando es de presentación tardía, después de los 65 años, vamos a ver que es una enfermedad poligénica, tiene más entre 60 al 80% del riesgo es hereditario, pero tenemos 1/3 que es modificable. Si hablamos de demencia frontotemporal, esto nos cambia. Vamos a ver que incluso la demencia frontotemporal esporádica, cuando el paciente dice nadie más en la familia había tenido esto, puede tener causas genéticas. Y cuando tenemos más de un individuo afectado en una familia con demencia frontotemporal, es necesario descartar una causa genética, porque de los individuos que tienen demencia frontotemporal familiar, que es más o menos el 30% de los individuos con demencia frontotemporal, la gran mayoría de ellos van a presentar variantes en alguno de los genes asociados a demencia frontotemporal. Van a ver que aquí hay una serie de números que determinan la frecuencia de presentación de los genes en MAPT, progranulina, C9orf, 72. Estos genes, es válido, es cierto, que están asociados con demencia frontotemporal, pero estas frecuencias de presentación corresponden a estudios europeos. Ok. Finalmente, vamos a ver que también, más allá de la demencia tipo Alzheimer, que tiene cierto grado de heredabilidad, la demencia frontotemporal que tiene mucha mayor heredabilidad, sobre todo cuando hay varios individuos enfermos en una familia, y finalmente tenemos demencias netamente genéticas. Tenemos la enfermedad de Huntington, Wilson, algunas leucoencefalopatías o lipofuscinosis del adulto, CADASIL, Fabry, neurodegeneración con acumulación de hierro, que aunque son muy infrecuentes, requieren una gran astucia por parte del profesional médico o profesional del área de la salud para determinar qué es una demencia posiblemente genética y que valdría la pena hacerle un estudio genético. Esto nos lleva a nuestra siguiente fase, que es, ok, si estamos pensando en mandar un estudio genético, ¿qué pacientes se beneficiarían de este proceso? ¿Qué pacientes son los que les debemos pedir estudios genéticos? Algo muy simple. Pensemos en edad de presentación. Paciente que tiene demencia antes de los 45 años, es casi que mandatorio buscarle una causa genética, hoy que puede ser incluso un síndrome metabólico. En pacientes entre 45 y 65 años vamos a ver que hay diferentes tipos de patologías que se empiezan a manifestar. Excepcionalmente tendríamos un individuo APOE4 presentándose con demencia temprana, es posible, pero vale la pena interrogar por la historia familiar. Más allá de la edad de 65 años, no es necesario, a no ser de que el paciente tenga una historia familiar muy fuerte de demencia precoz en otros primos, en otros familiares, y que, digamos, este paciente sea la excepción que se está presentando un poco más tarde. Pero en términos generales, vamos a ver que los diferentes algoritmos o las diferentes recomendaciones de cómo enfocar al paciente con demencia y determinar quién necesita un diagnóstico genético se enfocan en los siguientes elementos. Edad de inicio. Ya les dije, mientras más joven, es más importante el estudio genético. Presentación clínica. Si el paciente tiene demencia frontotemporal, es más probable que tenga causa genética que un paciente con enfermedad de Alzheimer. Si tiene antecedentes familiares de la misma enfermedad, mientras más parecidos sean los síntomas entre los diferentes miembros de la familia, es más probable que sea una causa genética única. Y si en una familia hay múltiples individuos con inicio precoz, inicio antes de los 65 años, amerita hacer un estudio genético. ¿Qué hemos hecho en Colombia? En Colombia comenzamos hace muchos años, más de 30 años, con el trabajo del profesor Francisco Lopera en Antioquia, en las familias de Yarumal, pero ese trabajo se ha ido enriqueciendo y se ha ido enriqueciendo a medida que hemos explorado diferentes áreas del país, diferentes comunidades, y hemos ido aprendiendo más de cómo la historia específica de nuestro país ha llevado a la coexistencia de muchas familias con enfermedad neurodegenerativa genética. Entonces, antes de seguir hablando de neurodegeneración, vamos a hacer un paréntesis para discutir la historia demográfica del país y de América Latina, donde todo comienza con las poblaciones amerindias, que eran muy diversas entre sí, y de repente tenemos una invasión española que luego comienza a importar individuos africanos esclavizados, Y estas tres poblaciones que estuvieron separadas durante miles de años se encuentran en el mismo espacio geográfico. Esto tiene como consecuencia el mestizaje, pero justo después de que estas poblaciones empiezan a intercambiar genes entre sí, a nacer nuevos individuos mestizos, tenemos un gran cuello de botella. Tenemos un cuello de botella porque la población se redujo de forma sustancial. Las infecciones, las guerras, llevaron a que solamente una pequeña parte de los individuos que estaban en el continente en ese momento sobrevive este cuello de botella y luego crece. Crece hasta representar más de 50 millones de colombianos que somos en este momento. Y a medida que crece, empiezan a llegar en el siglo XIX, en el siglo XX, olas de inmigración de diferentes países. Por ejemplo, en Colombia sabemos de los inmigrantes del Medio Oriente en el área de la costa, tenemos en Perú, en Brasil, inmigración del Asia, en Argentina, Chile, Brasil, inmigración de Alemania, Polonia y otros países de Europa. Estas condiciones son fundamentales para poder entender los genomas actuales de los individuos mestizos en Colombia y en América Latina. Y sobre todo, debemos entender este momento histórico, donde se genera un cuello de botella y tengo un número pequeño de individuos en nuestra población. ¿Qué pasa aquí? Estos individuos que quedaron después de las infecciones, después de las masacres, empiezan a reproducirse y empiezan a migrar hacia las diferentes subregiones, pueblos, las montañas, los llanos, etc. Y son pocos. Entonces, así hoy seamos 50 millones de colombianos, descendemos de un grupo pequeño que lleva a que si yo me comparo mi genoma con alguno de ustedes, siendo también de Colombia, puede que yo solo tenga que subir 6, 7, 8 generaciones hasta encontrar un ancestro en común. Esto es lo que vamos a llamar parentesco de fondo, donde así los individuos no se identifiquen como primos primeros, vamos a ver que comparten un gran número de ancestros en las generaciones recientes. Vemos el efecto fundador, que es cuando un grupo pequeño de personas se va y funda un pueblo y los descendientes son todos descendientes de este pequeño grupo de personas. Y la deriva genética es cómo este cambio de el número de individuos de una población va a afectar la frecuencia de determinados alelos o rasgos dentro de mi misma población. Si yo tengo, por ejemplo, un fenotipo muy excepcional, el cabello rojo, en la población ancestral, pero resulta que mi efecto fundador llevó a que solamente hubiera 20 personas, donde una de ellas tenía cabello rojo, a medida que pasan las generaciones, puede generar una población derivada donde el cabello rojo está presente en 10% de la población. Y eso lo explica ese pequeño tamaño efectivo de población, de la que se derivan todos los otros individuos. En adición, vamos a tener el mestizaje. Este es un ejemplo de una pareja, tiene 2 hijos, y en cada generación, a medida que ocurren eventos de mestizaje, empezamos a ver cómo el intercambio de material genético entre los cromosomas nos empieza a generar mosaicos de diferentes haplotipos, de haplotipos amarillos, verde azules o naranjado. Tenemos aquí un ejemplo de 2 individuos que heredaron el haplotipo amarillo del mismo ancestro. Entonces a este le vamos a dar el término de identidad por descendencia, porque este mismo segmento lo heredaron de un ancestro que tienen en común. Y si, por ejemplo, existiera una mutación que me causa la enfermedad de Alzheimer y que la heredaron ambos de dicho ancestro, no van a heredar solo la mutación, sino que van a heredar todo el haplotipo amarillo alrededor de la mutación estrella. La longitud de este haplotipo nos va a determinar hace cuántas generaciones estos dos individuos tienen el ancestro en común. Y este haplotipo lo podemos detectar utilizando métodos de secuenciación genética y un poquito de computación. En Colombia hemos identificado 13 variantes patogénicas en el gen de la preselinina 1. Estas variantes están a lo largo del país, considerando que son las zonas que más hemos trabajado. Y gracias a la posibilidad de estudiar el haplotipo alrededor de una variante o de una mutación genética, hemos podido identificar que estas variantes tienen diferentes orígenes ancestrales. Gracias a nuestra calidad de mestizos, hemos heredado variantes patogénicas en preselinina 1 de ancestros europeos, ancestros amerindios y ancestros africanos. Lo otro que hemos notado en Colombia es que las variantes genéticas, las mutaciones que nos causan enfermedades, se tienden a encontrar alrededor de las mismas zonas geográficas. Por ejemplo, estas 3 familias del oriente antioqueño tienen la misma mutación en el gen de MAPT y resulta que, aunque no se identifiquen como primos directamente, tienen un ancestro en común. Y si yo miro y comparo los haplotipos donde está esta variante en cada uno de los portadores, encuentro que todas las parejas de individuos comparten un segmento en común alrededor del gen que codifica para la proteína Tau. Entonces, hemos visto cómo este proceso de mestizaje, los efectos fundadores, nos están llevando a una carga genética característica de la población colombiana. Además de los genes mencionados de preselinina 1 y MAPT, hemos encontrado otra gran cantidad de genes causales de demencia o neurodegeneración genética. Cuando tomamos los genes causales de demencia frontotemporal, encontramos múltiples variantes en el país. A la hora de hacer el mismo proceso de determinar el origen ancestral de estas variantes, observamos que cuando tomábamos los genes causales de demencia frontotemporal reportados en la literatura, encontrábamos variantes todas en haplotipos europeos. Continuando en nuestra búsqueda, y como la cohorte de estudio incluía unos pacientes con esclerosis lateral amiotrófica, decidimos hacer el mismo ejercicio de buscar variantes que nos explicaran la patología de los pacientes con esclerosis lateral amiotrófica. ¿Qué nos llamó la atención? Fue que encontramos una gran variedad de mutaciones genéticas que en teoría causan esclerosis lateral amiotrófica. Estábamos viéndolas en pacientes con demencia frontotemporal y que estas variantes que esperábamos que nos causaran ELA no solo nos estaban causando demencia frontotemporal, sino que estaban en un haplotipo amerindio. Esto nos lleva a cuestionar si se hubieran incluido individuos de poblaciones diversas, por ejemplo, población latinoamericana, en los estudios de genética de la demencia frontotemporal o de genética de la demencia de la esclerosis lateral amiotrófica, posiblemente estos genes no se habían clasificado exclusivamente como causales de ELA, sino que pensaríamos que son genes también relacionados con demencia frontotemporal, que es el proceso de inclusión de individuos no europeos que lleva a este descubrimiento de este gen también puede estar asociado a este fenotipo. En los últimos años, desde el 2020, hemos estado expandiendo nuestro trabajo y colaborando con otros centros de investigación en América Latina. El proyecto de RedLab ha llevado a una gran colaboración y camaradería entre científicos de diferentes países, de diferentes instituciones, desde Estados Unidos, México, Colombia, Perú, Brasil, Chile, Argentina, para desarrollar una cohorte excepcional de individuos con enfermedad de Alzheimer, demencia frontotemporal y sanos en el continente. Vemos como la historia demográfica de cada uno de estos países nos genera un patrón diferente de mestizaje. Vemos, por ejemplo, en Argentina, en Chile, un componente europeo muy grande, lo mismo en el Brasil, y vemos que en Colombia, México, Perú, tenemos, por ejemplo, unos individuos con un gran porcentaje de componente amerindio, y en Colombia y Brasil tenemos individuos con un alto porcentaje de componente africano. Entonces, el patrón de mestizaje es heterogéneo, pero seguimos patrones relativamente similares entre los países. Vamos a ver que en América Latina, la principal causa de demencia frontotemporal fueron variantes en el gen de la progranulina. Las variantes en el gen de C9orf72, que se considera el más común, realmente fueron menos y fueron en los países que tenían ascendencia del norte de Europa. En Colombia identificamos un par de individuos con C9orf72, pero la mayoría de ellos venían de Chile. Lo más interesante dentro de todos estos genes que nos explican enfermedad de Alzheimer, como preselinina 1 y la demencia frontotemporal, fue encontrar una serie de familias con mutación en un gen que se llama tarde BP y me codifica para la proteína TDP43. Vemos un gran número de familias y familias muy extensas, principalmente en Colombia, estas 4 familias grandes. Tenemos estas 2 familias en Brasil que estamos explorando un poco más y vemos que este gen que en poblaciones europeas se caracteriza por presentarse usualmente como esclerosis lateral amiotrófica, lo estamos observando en nuestra población con un fenotipo supremamente heterogéneo. Vamos a ver individuos con demencia frontotemporal comportamental, ELA, trastornos del neurodesarrollo y individuos con variantes del lenguaje, afasia primaria progresiva, presentándose también dentro de las mismas familias que tienen, por ejemplo, un hermano con DFT comportamental, uno con ELA, un individuo con APP dentro de la misma familia, lo cual es supremamente interesante porque nos lleva a cuestionar cuáles son los factores que determinan que un portador de esta variante se presente con determinado fenotipo. Para concluir la charla de hoy, quiero hablar un poquito del riesgo genético. Lo que he estado discutiendo son, como les dije desde el principio, situaciones excepcionales, donde vamos a ver Alzheimer genético o demencia frontotemporal genética, pero la mayoría de nuestros pacientes tienen diferentes factores de riesgo ambientales, genéticos, y me gustaría aprovechar este espacio para discutir cómo debemos interpretar estos factores de riesgo con un poquito más de análisis que lo que inicialmente leemos de si usted tiene APOE4 va a tener X veces mayor de riesgo. Entonces quiero comenzar con el caso de TREM2. Este es mi primer invitado. TREM2 es un gen que me causa una demencia frontotemporal autosómica recesiva. En algunos casos, los individuos también desarrollan quistes óseos y recibe la enfermedad, el epónimo de Nasu-Hakola, pero también puede ser DFT sin compromiso óseo. Cuando no lo tenemos como enfermedad autosómica recesiva, sino que solo tenemos una variante en una copia, vamos a tener mayor riesgo de enfermedad de Alzheimer o de demencia frontotemporal. Traigo 2 variantes a la presentación de hoy. Esta la vimos al principio en el panorama del riesgo genético para enfermedad de Alzheimer y vemos que esta mutación aumenta el riesgo de enfermedad de Alzheimer en cohortes europeas. También se cree que puede aumentar el riesgo de demencia frontotemporal y tenemos la variante H157Y que se ha reportado en cohortes chinas, cohortes japonesas y diferentes metaanálisis donde los pacientes vienen del este del Asia. Estas dos variantes están presentes en Colombia. Vamos a ver que la primera la encontramos en un haplotipo europeo en los individuos colombianos y la segunda la encontramos en un haplotipo amerindio. ¿Qué es interesante de estas dos variantes? Primero, que la frecuencia poblacional, es decir, cuántos individuos tienen esta variante, es mayor en las cohortes americanas que en las cohortes europeas. ¿Qué significa esto? Que así como en mi ejemplo del cabello rojo, esta variante de TREM2R47H vino con los europeos, pero al tener una frecuencia baja y pasar por un cuello de botella, esta frecuencia poblacional me cambia. Y el número de individuos en relación de portadores versus la población completa, me cambió esta relación de portadores, no portadores y lleva aunque es una variante europea, en la población derivada es mucho más frecuente que en la población ancestral. Entonces, si vemos en las cohortes que se han reclutado en el proyecto de los 1000 genomas, en EXAT, en NOMAD, la frecuencia de esta variante en americanos mestizos es mayor que en europeos, aunque sea una variante de origen europeo. ¿Qué nos llama la atención? Que también tenemos esta variante que no está presente en europeos, no está presente en africanos, pero sí está presente en individuos del este del Asia y en latinos. Incluso en latinos es extremadamente alta. Y nos llevamos a preguntar, ok, ¿qué está pasando aquí? Ya hablamos de la historia demográfica reciente, ya hablamos del mestizaje, ya hablamos de los cuellos de botella, de la deriva genética, que es lo que me cambia la frecuencia alélica, pero ¿qué está pasando aquí? ¿Qué parentesco tienen nuestros individuos amerindios, que eran nuestros ancestros, nosotros actualmente que tenemos un componente amerindio y los individuos que el sol de hoy viven en China y en Japón? Bueno, resulta que si me devuelvo miles de años, voy a tener la migración fuera de África, donde el ser humano aparece en África y sale del continente y empieza a migrar. A medida que migras hacia el este del Asia, tenemos un pequeño grupo de individuos que van a subir, cruzar el estrecho de Bering y comenzar a poblar las Américas de norte a sur. Algunos de ellos se cree que tuvieron también contacto a través de la Polinesia, pero la principal ola migratoria fue los que venían desde el estrecho de Bering. Ya les había mostrado cómo podemos utilizar la genética para comparar individuos de dos regiones del oriente antioqueño o de la costa colombiana, pero ¿qué pasa si nos tenemos que devolver más en el tiempo? Tenemos otras formas que ya nos llevan en miles de millones de años y es comparar haplotipos portadores, no portadores y pensar que todos los haplotipos, si vienen del mismo origen, que es la migración humana, y lo que determina que sean diferentes son los diferentes eventos donde ocurren mutaciones, nos podemos devolver en el tiempo y buscar en qué momento los no portadores o los portadores comparten un ancestro en común. Entonces, si comparamos los individuos portadores de la variante TREM2H157Y en el proyecto de los 1000 genomas con los individuos portadores del este del Asia, vamos a ver que son parientes hace 31.000 años y que nosotros también heredamos a medida que los individuos amerindios migraron y llegaron desde el estrecho de Bering hasta Colombia y el sur de América, traían estas variantes consigo. Y ellos también pasaron por una serie de cuellos de botella que incrementaban cada cuello de botella nos fue incrementando la frecuencia de este alelo. Entonces llegamos a un escenario donde en nuestra población tenemos una interacción del parentesco reciente entre individuos de una misma área geográfica, el parentesco antiguo y la coexistencia de factores de riesgo multiancestrales, individuos que tienen variantes europeas, variantes amerindias, el mismo individuo puede tener ambas y aumentar el riesgo para su enfermedad de Alzheimer. Quiero terminar con el caso de APOE4. APOE4, como les mostré al principio, es el principal factor de riesgo conocido para la enfermedad de Alzheimer de inicio tardío. De hecho, APOE4 es la variante ancestral, la mutación es APOE3, pero se volvió mucho más común la variante derivada y está presente en todas las poblaciones. Vamos a tener APOE4 en todas las poblaciones, amerindia, europea, africana, el sur del Asia, mientras que es un escenario diferente al de TREM2, donde las mutaciones de TREM2 se observan aún aisladas entre las poblaciones ancestrales. ¿Qué pasa con APOE4? Vamos a ver que el efecto que tiene este gen o este genotipo es diferente según la población. Tiene su mayor riesgo en individuos asiáticos, por ejemplo, en individuos japoneses, tiene un 30%, un 30% de odds ratio, menos en europeos, un poco menos en afroamericanos y un poco menos en africanos. Esto es bastante interesante y llevó a los investigadores a cuestionarse qué es lo que lleva a que la misma variante, la misma mutación, nos genere diferentes patrones de riesgo en las poblaciones. Es el estilo de vida, es la interacción con otros genes, y hablando de, es porque tienen ancestría asiática, ancestría europea, pero fue supremamente interesante cuando el estudio de individuos mestizos que eran portadores de APOE4 nos permitió subclasificar los individuos mestizos portadores de APOE4 en APOE4 derivado de poblaciones europeas, derivado de poblaciones africanas, poblaciones amerindias, y nos llevó a entender que era el haplotipo específico de APOE4, no solo el gen, no solo la mutación, sino ese fragmento que al principio veíamos alrededor de la estrella en amarillo, como ese fragmento de otras mutaciones, otras variantes que se están coheredando con APOE4, modulan o modifican el riesgo que esta variante nos está generando para la enfermedad de Alzheimer. Entonces, si yo quisiera tomar esas imágenes que mostré al principio del riesgo genético para enfermedad de Alzheimer, todos los valores, o bueno, no todos, pero los valores de riesgo y de frecuencia poblacional se me moverían de acuerdo a las diferentes poblaciones. Entonces, en conclusión, es fundamental conocer una población y un poquito más de la historia de dónde venimos para entender cómo el riesgo genético, la carga genética es única y específica para la población latinoamericana. La carga genética está intrínsecamente relacionada con la historia demográfica y esto es importante a la hora de diseñar, interpretar estudios genéticos y nos lleva a insistir en incluir individuos mestizos en los diferentes estudios o los diferentes esfuerzos globales para entender la demencia. Entonces, mil gracias. Este trabajo tiene muchísimas personas que colaboraron para que lo pudiéramos presentar, para que pueda existir. Principalmente hoy le quiero dar gracias al doctor Ken Kosick, a la doctora Diana Matallana, al doctor Francisco Lopera y a todos los individuos que participan en el proyecto de Red Lab. Entonces, muchísimas gracias. Estoy disponible para preguntas.
Muchísimas gracias, doctora Costa. Me gustaría saber si alguien en la sala tiene preguntas. Tenemos una sola pregunta para hacer. Gracias, doctora, por su tiempo. ¿Alguna pregunta? Hay una pregunta. Esperamos que sea corta.
Buenas tardes, doctora. Muchísimas gracias por la charla, muy interesante. A mí me llamó mucho la atención lo que comentaba sobre esos genes que comparten la enfermedad de esclerosis lateral amiotrófica y la demencia frontotemporal. Entonces, mi pregunta tiene dos partes. La primera es si eso pasa con otras enfermedades también, por ejemplo, la enfermedad de Alzheimer y la demencia frontotemporal, o con la enfermedad de cuerpos de Lewy. Y la otra es cuáles serían los factores que determinan que se presente pues clínicamente una enfermedad o la otra. Es decir, si tengo los genes que comparten ambas enfermedades, entonces, ¿qué determina que sea frontotemporal o ELA en el caso que tú nos mostraste? Gracias.
Listo. Yo creo que la respuesta a tu pregunta tiene que ver con un término que en genética llamamos pleiotropía, y es que un mismo gen puede tener diferentes manifestaciones. Lo importante acá es que es muy difícil tomar un gen y observar las manifestaciones, porque nosotros no podemos hacer experimentos de mutagénesis en humanos. Entonces, solamente observamos las manifestaciones y a partir de estas observaciones de los fenotipos, concluimos los genes. Entonces, en humanos solamente podemos hacer esta dirección. No podemos inducir mutaciones para ver los fenotipos. Eso se puede hacer en otros modelos animales, pero que a la hora de tratarse de enfermedades cognitivas no son los mejores para explicarnos qué está ocurriendo. Entonces, este efecto de pleiotropía, donde un gen puede asociarse con múltiples fenotipos, lo observamos en enfermedades que tienen sintomatología en común. Por ejemplo, va a haber casos poco frecuentes donde un paciente con mutación en preselinina 1 asociado a demencia de la enfermedad de Alzheimer tenga una manifestación fenotípica de demencia frontotemporal, o un paciente, a la inversa, con una mutación en el gen de tau, en el gen MAPT, tenga un fenotipo de enfermedad de Alzheimer. Entonces, la pleiotropía nos afecta a muchísimos genes de patologías que están relacionadas ya sea desde la sintomatología o desde los mecanismos moleculares que tienen en común. Ahora, ¿qué lleva o cómo podemos predecir que un paciente va a presentarse como una ELA o va a presentarse como una DFT? Aún no sabemos. Es un área activa de nuestras investigaciones. Aún no tenemos explicación de qué lleva, porque si piensas, por ejemplo, en las familias que te mostré, hay individuos que son hermanos y al ser hermanos comparten una gran cantidad de ADN, pero no sabemos por qué un hermano tiene ELA, un hermano tiene DFT. Aún no tengo respuesta para eso.
Doctora Costa, muchísimas gracias por su presentación, muchas gracias por su tiempo. Fue un honor tenerla. Un aplauso para la doctora Costa.
Muchas gracias.
Muchas gracias. Now we continue with Dr. Natalia Vilor Tejedor. She is a senior researcher at the Barcelona Beta Brain Research Center, where she leads the Alpha Genetics Project and is a member of the Computational Biology of RNA Processing Group at the Center for Genomic Regulation, CRG. She holds a bachelor's degree in mathematics and applied statistics and a master's degree in omics and data analysis, and a PhD in biomedicine from Pompeu Fabra University. Her research focuses on identifying genetic risk and protective factors for neurodegenerative diseases by integrating omics data, neuroimaging, and transcriptomic analysis. Welcome, Doctor. Doctora, ¿tienes tu micrófono apagado?
Yeah, sorry, it was not working. Sorry. Okay, I think I'm sharing already my screen. And probably you are seeing all the presentation. So yeah, buenas tardes, good afternoon, good evening to everyone. Thanks a lot for this nice introduction. And also thanks a lot to the organizers for the invitation. It's a pleasure to be here presenting at this first Colombian Brain Health Congress. I will be presenting some of the work of my team that is strongly related with the topics of this Congress. And this work is mainly focused on genetic epidemiology and statistical modeling in the context of brain health and neurodegeneration. I will cover some topics that were already discussed in the previous presentation, and I think it's very nice, all of these links and how we can put all together for this prevention and clinical translation of Alzheimer's disease. So basically what we are doing in our team, or we are interested, are in these risk factors that contribute to brain health and disease, especially neurodegeneration. So mainly our main research question or objective is to try to understand which are the variants, the genetic variants that are related to this brain health or to these neurodegenerative diseases. And in a direct approach, it could be sufficient to address this association or relationship, analyzing how these variants connect with these case control studies, for instance, in Alzheimer's disease. The problem is that when we define these cases and controls, we have some problems. The first one is the heterogeneity of the disease, but also the misclassification of cases and controls using this clinical definition. And this is why what we are doing in our studies is to try to connect these genetic variants with brain endophenotypes, with brain measurements that are considered better targets for genetic analysis. Many brain-related traits exhibit a significant degree of heritability, and this indicates a substantial portion of the observed variability in these traits attributed to these genetic factors. So this is a very important point. The other thing is that these measurements have a strong biological base, and therefore these genetic variants might affect the brain features more directly, as I was saying, than the clinical definition of the disease. And this is actually good because we can have these intermediate phenotypes, our brain phenotypes, between this connection of genetics and the disease. But also these brain features are genetically correlated. And this is also why we can then connect the factors that we are finding in this analysis with the proper disease. Somehow we can figure out how, at the end, one factor that is associated to a volume can be affecting also the progression to this neurodegeneration, or in our case, Alzheimer's disease. And this is also important because if we analyze our brain endophenotypes, or we use these brain endophenotypes as our outcomes in our analysis, we're going to require a smaller sample size because of this direct genetic effect on the influence of the brain. Another important thing is that, for instance, in our case in Barcelona, we are working in a cohort that is still cognitively unimpaired. So we don't have this definition of clinical status, but we have these brain measurements. We know that our brain starts to, or the neurodegeneration starts to appear years before the development of the neurodegenerative symptoms. So this means that even in cognitively unimpaired individuals, we are able now to analyze how genetic factors affect the brain of this person, and then how these can connect with a future progression of the disease. So all of this creates a nice environment that allows preventive measurements years before the appearance of the symptomatology of neurodegeneration. And this was actually nice to visualize, and some big consortia started to analyze and to perform some big analysis to try to understand which variants were connected with some cortical structures. In this study, for instance, this is one of the first ones that was published. Hibbard and colleagues identified several genetic variants that were related to different subcortical regions. For instance, here in the putamen, they found the most significant hit, but also in the hippocampal region, and so on. And in every of these studies, what we are trying to do is to first increase the sample sizes and also to be more precise with the brain features that we are analyzing. Another study was this one that was published actually 5 years ago. And here what we were trying to understand was the association between genetic variants and the human cerebral cortex. And also here, this was one of the first studies finding around 200 hits for each of these features. And this is important. But as I was saying, we are on one side trying to put efforts on increasing these sample sizes, contributing with different cohorts around the world. But the other thing is to try to be precise, thanks to the technology that we have now, for uncovering and for acquiring more precise regions related to our hypothesis. For instance, in my team, what we are trying to do is to develop new acquisition methodologies or applying new methodologies for trying to disentangle, for instance, some cerebrovascular characteristics. One of the main regions or measurements that we want or we are interested on are the perivascular spaces that are very difficult to quantify. We try actually to quantify this manually, thanks to a radiologist that was in our team. This means that this person needed to manually, visually evaluate around 1,000 images and quantify all of these counts of the perivascular spaces. This is a huge work, but thanks to this, we were then able to, for instance, perform this nice study analyzing how the genetic variants associated to Alzheimer's disease were related to this count of perivascular spaces in two different regions, in the basal ganglia and in the centrum cerebral. And what we found, here you can see in this panel, is that in ApoE4 carriers, those that also were carriers of this BIN1 variant that is also strongly related to Alzheimer's disease, the second most significant one, were at increased probability to present perivascular spaces in this centrum cerebral region. What we did after, thanks to also other colleagues and people from the team, was to use a method, a deep learning algorithm that was developed in the Erasmus Medical Center and was published in 2019. So thanks to this algorithm, we were able to quantify the perivascular spaces automatically in 4 different regions, 2 more regions than manually. And actually what we are doing now is to try to understand what are the genetic but also characteristics that influence the appearance of these perivascular spaces. You can see here some interesting patterns based on age and sex. Actually, we only found significant differences by sex in the hippocampus, although here you can see the early ages that there's some difference as well, but not based on age. And we can actually explore better the preliminary results that we found. Here we have the 4 regions of where we quantify the perivascular spaces. We stratify actually for each of the factors, risk factors that were significant for at least one of the regions. We stratify the models by amyloid status. Here, each bar represents amyloid negative, amyloid positive. And actually, if you see here the results, the most interesting ones are in the hippocampus. And actually, the 2 most interesting for me are the ones that are happening here in the sex variable, but also in the ApoE carriership. So basically, what we found is that ApoE4 carriers at this region are more exposed or have more probabilities to develop these perivascular spaces. And this is actually very interesting. Another thing that we want to try to uncover are how genetics influence the trajectories of some brain regions. Here, for instance, in this study, what we did was to quantify the trajectory of the hippocampal volumes and some hippocampal subfield volumes that are available in the ADNI study. We have around 9 time points, so 9 different measurements. And we were able first to see how the trajectories of these different hippocampal regions based on the, let's say, aging, so how the trajectory is evolving for the different disease stages that we have available in acne, controls, MCIs, and ADs. And here we can see the aging pattern, no, very clearly. We can see how the atrophy is happening based on because we are getting old, and also the difference between being a control or being an AD. But what we wanted to do is to try to understand if the genetics of Alzheimer's disease, so the genetic risk of Alzheimer's disease, was accelerating this atrophy. And we performed some different analyses. Here I'm just showing this figure that summarizes a bit the results. We performed some linear mixed effect models for analyzing how the genetic risk of Alzheimer's disease quantified through a polygenic risk score interact with time and also with disease status. And what we found is basically what you are seeing here, that being a APOE4 carrier, but also being at high risk for Alzheimer's disease genetically, was influencing these trajectories. And the most interesting part is when we were also stratifying the results of the interactions, we found that these results were driven by these MCI individuals. So basically, the genetics is affecting the trajectories of the hippocampus regions, or the atrophy of the hippocampal volume, in this stage mainly, which is very interesting for a nice discussion. Another thing that we are trying to understand in our team, or what we want to do, is to link also environmental factors to brain health and to Alzheimer's disease and dementia. And we want to do that. Mainly we are focused our analysis on air pollution first, because since 2020 and now in 2024, in this Lancet paper, for the first time in 2020 is when air pollution appeared here as one of the risk factors for dementia. And this was also in 2024, increasing actually the percentage of risk attributed to air pollution. So what we wanted, or what we were exploring, is to try to see, okay, how many cases of dementia can we prevent if we modify the risk factor, and also are we able to link air pollution not only with dementia but also with Alzheimer's disease? This was our two questions as well in the teams. So the first thing that we did was to try to understand the connection between air pollution and AD pathology. And for this, in our sample, in the Alpha study in Barcelona, we have some CSF biomarkers and also some PET data. And what we found is that actually air pollution was associated with NFL in CSF, but also with amyloid beta deposition. And if you see here also in these panels, we also found that these effects were driven by amyloid positive. So only the results were significant in amyloid beta positive individuals, which is suggesting this link as well between air pollution and AD pathology. A second project that we proposed was to try to understand a bit better linking air pollution with other CSF and plasma biomarkers that we have available, for understanding a bit the pathways through how air pollution is affecting this risk of dementia and AD pathology. And what we found was mainly a strong association between air pollution and GFIP. As you can see here, basically air pollution was affecting the levels of GFIP and increasing the levels when the person was exposed to higher levels of pollution. And interestingly as well, when we stratify this analysis by sex, we found that only in women these effects remain significant, which is also an important point in our studies, these sex-specific differences. We then also further analyze how this biomarker, GFIP, was mediating the relationship between air pollution and some cognitive measurements. And what we found, also only in women, is that there was an indirect effect through which air pollution was affecting executive function and attention by modifying the levels of GFIP. So there's a direct effect, a relationship between air pollution and cognition. We saw that in previous studies that we published as well. But here, for the first time, we saw that there's an effect of air pollution and cognition through this path, through the modification or the increase of levels of GFIP. And this is also a strong and a new result that is linking air pollution as well with neuroinflammation that perhaps can be also influencing these dementia or AD processes. What we are doing also now is to try to understand a bit better, not only with the effect of air pollution and biomarkers, but also with the effect of air pollution and brain features. We want to cover all the possible intermediate phenotypes or endophenotypes related to Alzheimer's disease and neurodegenerative process. So we wanted to propose also this study, and I'm gonna show a bit better. So here what we did was to also compute some polygenic risk scores for also reducing the number of genetic variants that we wanted to analyze. And we proposed some interaction models with the different pollutants that we quantified as well, and these genetic scores related to Alzheimer's disease. And here you have a summary of the results that we found. These are the different regions, here, for instance, from the hippocampal region, and here from some cortical and subcortical brain regions. Here I'm showing only the significant results and the p-value for the interaction. And what we mainly found was a strong effect of the risk of Alzheimer's disease, but also the risk of a score that means or represents a proxy of inflammation. And mainly what we saw is that if we focus, for instance, in the hippocampal regions, there's a strong effect of this a strong interaction between this exposure, this global exposure to pollution, and this proxy of inflammation in several hippocampal subfields. Only one when we consider the genetic risk of Alzheimer's disease. Perhaps we were expecting more, but I think it's okay. But then if we go to a more general definition, or when we use the cortical and subcortical brain regions, here there's actually a bit of overlap. We see effect more on the temporal regions related to this interaction with AD, with the genetic risk of AD and exposure, and here more a temporal and parietal affectation when we consider the genetic proxy of inflammation in interaction with the exposure. And in all the cases, if we also see a bit the results stratifying by high-low genetic risk, here, for instance, I took this example, the transverse temporal volume, we can see actually that in all cases, we here see the trend, all the individuals that are at high genetic predisposition of inflammation or of AD, when are exposed to a higher level of pollution, the volume of the region is reduced. And this is actually the pattern that is happening in all the results that I was showing before. And also we wanted to understand a bit better, so not only with this proxy of genetic predisposition to Alzheimer's disease or inflammation, but also to a more specific pathway definition. So we computed something that is called pathway PRSs of Alzheimer's disease to understand a bit better these biological signatures that define the genetic risk of Alzheimer's disease. And for instance, if we consider the same region that I was showing before, here we can see that actually the effect that we found is not driven, for instance, for this amyloid biological signature, but more from the kind of cholesterol complex lipoprotein signature or external stimulus, or even the immune system signature. So this is also important for understanding a bit better the biological mechanism and not only the significance of the genetic effects. And some other things that we are starting to perform are the inclusion of other molecular and environmental measurements on this interaction of models. So for instance, we are working with telomere length data. That is also important molecular marker of longevity and neurodegeneration. And also we are including other factors like fatty acids or omega factors that also in interaction with genetics, telomere length, or other factors can affect our brain health. But as I was saying at the beginning, our objective is on one side to try to understand the genetic and environmental mechanisms that are linked to this brain health. But on the other side, we are interested on understanding or improving the statistical models that we are using for predicting this neurodegeneration. And this is actually important because, well, as I was also showing, we can continue increasing the sample size and performing also always GWAS analysis or brain whole genome analysis. And yeah, if we increase the sample, we are gonna have more chances to identify more variants. But there's also, in some cases, scenarios in which, depending on the population, depending on the characteristics, we cannot find so many individuals. So we need also other strategies, and this is why we also are trying to propose some other statistical strategies for increasing this statistical power and being able to identify also factors related to our brain health and dementia. And some of the strategies that we propose are, for instance, gene set analysis strategies, so combining the genetic variants depending on the target gene in which they are included. Also, we are now assessing this computation of polygenicity, computation of polygenic risk scores, and pathway polygenic risk scores for uncovering a bit the mechanisms and biological pathways that are behind the associations that we are finding. And this is in collaboration to several cohorts and institutions. And here, mainly what we are trying to do is, when we combine this information, the genetic information, we try to see, as I saw before for the environmental data, we try to see the relationship between these genetic components or scores with brain outcomes or cognitive scores or other biomarkers. But also another thing that we are starting to also address is the information, the huge amount of information that we have available. We are talking about genetics. We also are talking about brain imaging. We also have available proteomic data. We have available other types of omics data. So at the end, we require or we need methodologies that combine all of these analyses to jointly analyze and to jointly understand all these modalities in an easy way. So this is why we are also developing this multivariate and multiphenotype analysis to try to combine all of this information. For instance, in this first study, we developed an algorithm that is an extension of a multifactorial analysis. And the idea was to retrieve latent factors that then were associated with outcomes of neurodegeneration. And in the second study, for instance, what we tried to do was to jointly analyze all the regions of our brain, so how genetics were affecting not univariately each of the regions, but as a component, as a big component. So if I am at high risk of Alzheimer's disease, how is this affecting my brain as a component, not univariately each of the regions that I have. Other things that we are proposing are, for instance, the improvement of the scores or the improvement of the capacity that we have for predicting neurodegeneration and aging. And here is very important also the concept of all this integration of omics data. We have available now a lot of information in our cohorts, but also in some public spaces. So this is very interesting because this allows to improve the score that we are using. For instance, for genetics, what we are trying to do is to retrieve some functional variants, PQTLs or EQTLs, that are genetic variants associated to changes in protein levels and gene levels. These are more informative sometimes than those that are retrieved from GWAS analysis, because at the end, we are selecting in the summary statistics of the GWAS those that are more significant based on a p-value, but a p-value doesn't have any biological meaning. So here what we are trying is to try to keep those that have actually the biological meaning and try to incorporate them in these scores to see if we are able to predict these common scores that are used based on this functional information. And then, of course, if we are able to do that, our interest is to try to see if we are also able to identify novel associations, relating them with our brain outcomes, our biomarkers, and also our cognitive outcomes. Other things that we are trying to improve is the incorporation of non-parametric tests in these genetic associations. Why? Because, for instance, in some of the studies, or for some of the measurements that we are using, we cannot assume a normal distribution. If we talk about volumes, yeah, that's true. But if we, for instance, work with other cerebrovascular measurements like white matter hyperintensities, and you check the distribution, this is not going to be normal. But anyway, a lot of researchers are using this approach. This is okay, but we are removing some probabilities to identify genetic variants that are actually true variants related to these features. So what we are trying to do is to develop and to adapt methodologies that don't assume any distribution for these outcomes, for these brain outcomes, that this can be applied also for other outcomes, and try to identify and see how can we improve the identification of this variant variants by using these non-parametric tests. And we're starting to apply this first in the context of brain imaging genetics, so genetic variant related to the brain. But this, if we validate this, this can be applied to other contexts when we don't have these normal distributions. Another interesting thing is to follow a similar strategy than the incorporation of genetic variants in one score, but using the environmental factors that we have. This is more a multimodal approach, and I think it's also very interesting because at the end, when we try to analyze all the potential confounders or lifestyle factors, environmental factors, I mean, there's a lot. And when we start to analyze all of them, we need to then penalize our models for multiple comparisons. All of this, it's interesting, but can be improved as well. And this is what we are trying to do, to try to create this score that weights the different exposures and merge them into a score. And this can actually improve then these gene environmental models that we are trying to apply. We can, at the end, summarize several models in just one model, one with a score that summarizes your genetic predisposition to Alzheimer's disease, for instance, another one that summarizes your exposure. And with this, we really think that we can find more interesting associations also with our brain health outcomes. And finally, I think, and this is very interesting, it's also a novel or not novel thing, because we all know machine learning and artificial intelligence, but I think now it's time also to adapt all of these methodologies for disease stage classification, for also latent factor and feature extraction. So we are also starting to analyze and to use these methodologies from using several approaches. And I think this is actually not the future, but the present. We are finding some interesting results with some pilot analysis that we are performing also in collaboration with other colleagues. And for the integration of all these layers and multi-platform data that we have, this is going to be very, very important. And as we are starting to see with all these feature extraction and classification methods. And finally, I think this is also very, very important because at the end, if we are trying to understand how risk factors contribute to health and disease, and we are also trying to improve our statistical prediction for prevention and for these strategies, we need to also translate the results into a more clinical aspect and also to try to be diverse and inclusive with our science. And this is actually things that we are also doing, trying to focus our analysis on a more diverse and inclusive way. And this is actually one of my last slides, because I wanted to show some results that are in collaboration with so many researchers, mainly in University, where we are trying actually to improve this diversity from different perspectives. So on one side, we recently published this paper that was led by Tavia Evans from University, where we show actually, we analyzed six different cohorts, several individuals, and we were exploring actually some patterns from MRI scans that were differing between these different individuals from different ancestries. And actually here, interestingly, we found that the major driver of this difference was ethnicity, more than some patterns of the proper MRI scans. So I think this is a huge work and a very interesting work that is also related with the previous talk. And I think we should continue working on this area. Another thing that we are also doing in collaboration with Aram and Tavia from Radboud is the development of these deep learning models for trying to quantify or extract some measurements from CT scans and also some low-field MRI scans for recovering the image in a quality that can be used for research and also clinical practice. And I think this is also a very, very important thing that is being led by Tavia and also by the United Consortium, and that should also require, of course, more data and also more diversity for trying to see if this is also something that can be adaptable to different populations and not only the ones that we are using now for this quantification. So if anyone is interested, I think it's an important contribution that we can all join force. Another interesting aspect that was also covered in the previous presentation is the role of the genetic ancestry and specifically the local ancestry. So we all know that there's these differences, was very well presented before, but also here what we wanted to also disentangle is what is happening at the haplotype level. And specifically, what is happening at the haplotype level of some lodges that are related with Alzheimer's disease. And our hypothesis is that probably some of these differences that cannot be seen if we assess this global view or this global genetic ancestry can affect somehow the associations that we are performing with some of our endophenotype brain health outcomes. And this is something also that we are starting to analyze and that we are really enthusiastic because we think there's going to be so interesting results, and it's something that I think we should all put some efforts on that. And finally, I was talking about multiple layers of diversity, and here I was presenting first the data, diversity in the data, but I always like to mention that for being diverse, we need to be diverse also in the hypotheses and in the people that is making the questions and the research. And for this, we need diverse also on the researchers that are working with the data. And this is actually something that we are trying to develop or to help in the Prisma Association. These are two projects that we got last year, that are actually for trying to disentangle first the gaps that are making, the challenges that are not making biomedicine inclusive. This is one thing. And the other thing that we are trying to understand are the biases that are in these deep learning and artificial intelligence algorithms that are very important because we are going to start to use a lot all of these. So we need to try to understand how are we biasing and how to correct these biases in our models. So with all of this, this is a bit the summary that I wanted to show you during the presentation. I'm happy to have your questions, to answer your questions. And the last thing is to thank you all for the invitation, for your attention, also the study participants and all the volunteers that we have. And this is why we can make our work and our projects, and also to my team, institution, and all of you. Thank you.
Thank you, Dr. Vilar Tejador. We have some questions from the audience. Okay. Perhaps you're done. I was very mesmerized by your presentation about all those methodologies that you presented, those cutting edges forms of measuring these ways in the environment affects genomics. So thank you very much. If anyone here wants to talk to you about ideas and these calls that you made, how can we reach you?
Sorry, because I cannot see you now.
Sorry.
I closed my presentation, and perhaps I can share my email and contact again, but I cannot do it now. But feel free to share my email and contact. And I mean, I would be happy to discuss with anyone about potential questions, also collaborations. We are very open to try to increase the diversity also in our analysis. So yeah, it would be great to have discussions and a bit of time for talking with everyone that is interested.
Thank you. Thank you very much. Please.
Thank you.
Thank you. Up next, we will have a little brief coffee break. And next, we're going to proceed with our seed grants and PhD-funded fast presentations. Dr. Coley, I think that couldn't make it today, so we're going to try to reschedule his presentation. We see in about 20 minutes. We will let you know. Sandrita, ¿me escuchas? ¿Me oyes? ¿No está en otro link? No, porque está acá. ¿Cómo va a ser otro link también?
Hola, yo sí les digo.
Ah, perfecto, ya. Igual te iba a comentar que vamos a tomarnos 5 minuticos más mientras le damos espacio al doctor Crowley para ver si podemos continuar con su presentación y si no arrancar con esta, para que lo tengas ahí también pendiente tú y los asistentes.
Ok, está bien. Dale, perfecto, aquí estoy pendiente.
Perfecto, gracias.
Welcome back and thank you. We're going to have now our final session of the day. We will be having PhD and SID grant awarded fast presentations, and that activity is going to take like around 2 hours. Our first presentation is in charge of Sandra Castelblanco. She is a doctoral student and she is a geriatrician. And Sandrita, we are all with you. Good luck.
Yes, thank you. Okay, can you see the slides?
Sí, sí los vemos.
Laura, can you confirm
Sí, sí las estamos viendo.
that? Can you see the slides? Okay, perfect. Okay, thank you very much. Thank you for being here. My PhD project is about structural and functional brain markers in MRI and factors associated with different levels of brain health.
Excuse me,
Well,
Sandrita.
I know that
The presentation is not on
okay.
on full screen.
Okay. Give me 1 moment, please.
Don't worry.
Okay, now
No, not yet.
it's in full screen. Okay. Okay. Let's see. Okay. And now?
No, not yet. I think you have to share your full screen.
No, yes, I'm looking my full screen.
No, pero como compartirla desde el inicio en el Zoom. Aunque si el público no tiene problema, creo que también verla así. Sí, no hay problema. No, podemos comenzar así igual. Dale, si quieres arranca. Disculpa.
Okay, but no, this is not the best option. A ver, ahí, can you see better?
No, es que estamos
I
viendo
don't
solamente
know
la pantalla de
why.
PowerPoint. Por eso creía que tenías que
Yes,
salirte
yes.
y volver a compartir.
Sí, yo te entiendo, solo que yo estoy poniendo presentación con diapositivas completa. Y no sé por qué no. De hecho, ahí tú estás viendo que pongo presentación desde el principio.
Sí, correcto.
Okay.
No, arranquemos así,
Okay.
si quieres, para no quitarte más tiempo.
Yes. It's okay.
Listo, go ahead. With you again, Sandra Castelblanco.
Okay, thank you very much. As I said, this is the title of my PhD project, is Structural and Functional Brain Markers in MRI and Factors Associated with Different Levels of Brain Health. I know that you have been spoken about a lot about brain health, but it's important to know that in this research, I adopt the definition of the concept of brain health that use the World Health Organization, which encompasses cognitive, mental, physical, and social well-being at every state of life, allowing individuals to analyze their full potential over life's course. But it is important to know that brain is one of the most important organs in the body because it helps us to control behavior, social emotional control, movement, solving problems, decision-making, communication, but also other basic functions like breathe or control autonomy functions. So these functions, the brain functions, help us to develop a role, a social role in our society. But there are some people who have better performance than others in cognitive and functional skills. So maybe there are some levels of brain health. It's just like there are people who have better or regular or more standard levels of brain health. So in this research, I use three levels of brain health. The general brain health, that is the stage in which the brain function effectively allowing individuals to reach the full potential with standard performance, and optimal brain health, that refers to the pinnacle of cognitive and functional performance, allowing individuals to adapt effectively to their environment and excel beyond normal patterns. And finally, brain health deficit, which is the impairment of cognitive and motor function, which is related to biological factors such as neurodegeneration or structural brain alterations. And in this research, I will use a special model which includes a cognitive function and physical function, and also mental health as a control to develop the models of the three groups. So cognitive function is evaluated with mental test. Physical function is evaluated with TDALQ test, and the mental health is controlled in all the groups. So all the groups have absence of self-reported mental or physical illness. And those are the points of the cut points to this each group. The aim of this project is describe structural markers, brain network connectivity in MRI, and cognitive, medical, and social factors associated with different levels of brain health in people in Latin America. This is important because you know that Latin America have a lot of challenge in their communities, such as increased dementia prevalence, a lot of barriers to achieving life goals, limited education opportunities, unequal healthcare access, high disease rates, and complex social conditions. But also Latin Americans have some protective factors which are different in other places of the world. These factors are community support, resilience and empathy, and cultural practice. And this study is part of an answer of the call who did the World Health Organization to optimizing brain health studies around the world. The methodology, this is a cross-sectional analytical observational study. This study used the control population red blood study, and I include adults over 50 years, Latin American residents, and without mental or brain health or brain disease. Also, I made the analysis of functional and cognitive performance characteristics to create the groups, and also to describe the characteristics of the three groups. Now I am working in structural analysis, but also I will work in functional imaging analysis using special tools to make this analysis of MRI. And with these results, I will create the regressions models to related cognitive factors, medical factors, and social factors with MRI markers results. These are some of my preliminary results, and these are the characteristics of the three groups. There are 257 subjects. There are no difference between sex, violence exposition, lung blindness, number of comorbidities, but there are differences between age, education levels, and of course, the performance of neuropsychological tests. And the voxel-based morphometry results, this is some preliminary results. This is a t-test which can difference the differences between areas of brain areas who are different between the three groups. In blue, you can see optimal brain health and contrast with brain health deficit, and in yellow you can see optimal brain health in contrast general brain health, and this is adjusted by FWE correct and using 15 voxel clusters. And as if I adjusted to volume, intracranial, age, education level, and sex, there are some areas, especially left and right amygdala, right hippocampus and parahippocampal regions, who are different between the three groups. And future results, well, then I have to determine the association between structural markers in brain health levels with social and clinical factors in Latin America. And finally, I have to calculate the functional connectivity in resting states networks in the three groups, and the results of this will be a part of models of regressions with social and clinical factors in the people from controls of red blood study. Thank you very much.
Thank you, Sandra. We have any questions from the audience? We have one.
Thank you so much, Sandra. And my question is about if you think that here Nano Santa Maria, just in case. Sandrita,
Yes, I know.
just to know your opinion about if you could like extrapolate or create a similar model for mental health, like the optimal brain health. I was wondering that probably you could have like something like, I don't know, optimal well-being or optimal mental health. And if that is possible, I don't know, I'm interested in just in your opinion, if that is possible, what kind of methods of strategies you have in mind for, I don't know, dealing with that?
Well, this is possible. Well, there are some things that I controlled to develop my model of brain health levels. For example, I didn't include the mental disease, but brain health is an integrative concept. So if you are speaking about brain health levels and an optimal brain health level, it could be a correlation with optimal mental health. I think that we can make more analysis about that. Maybe using the psychiatric skills that red blood use to make if there are a relationship with cognitive, but also with brain health levels. It could be an option. I think that maybe it's a good idea, so I will think in how to do that.
Thank you, Sandra. Any more questions? Okay. Thank you. Now we move to Laura Moreno Hernandez, another of the PhD students. The purpose of this session is to present all the PhD award by the D43 grant. So if you can give us any of all the questions that you have, it's very important because it promotes and boosts our own projects. So these are PhD students in different years. So Sandra, for example, it's in the last year of the PhD, and now Laura, who is in the third year. Yes, please welcome her. Round of applause. She has been also our co-host.
Well, good afternoon. My presentation, my project is about the relationship between brain health, perceived exclusion, and social determinants of health in Colombian women. Basically, this project comes from the idea that women in Colombia have a unique set of situations like ethnicity, poverty, and even exposition to violence that put them in a particular situation in which like their health can be affected, like their brain health can be affected. And we know that like the global tendency is that women have poorer brain health than men, even when they are older in a tendency. So this is the big situation we find. So we know that gender violence, gender stereotypes affect even the access to health services, and that's why we are like interested in seeing how these set of factors can affect particularly the brain health in women. So that's the idea. Well, what is the present situation, or what is the problem that we find. Basically, there's limited evidence on intersectionality. For example, the exposition to violence combined with the ethnicity, combined with the gender, and combined with other like social determinants of health. So I would like to know how these adversity patterns affect the brain health. So my aims are, well, the big goal is to understand how perceived exclusion impacts this brain health, as I've been repeating the whole presentation, I'm sorry. Then how this intersectionality is particular to Colombian women, that's why we want to characterize it. And then assess the association with mental health outcomes, because we are understanding that brain health, as the Prof. Hernando was asking, well, we know that mental health is is part of the brain health. So we want to assess this association with mental health outcomes. And also we want to compare how neural patterns have different expressions in relationship with the exclusion levels that we can see. So what are the hypotheses? The first one is that multiple disparities worsen mental health outcomes. I mean, being a woman is one of these part of this vulnerability, but then we have ethnicity, and then we have poverty, and then like it would be about, at least in my idea, is that it's going to be increasing the profiles of risk. Then higher scores in perceived exclusion will negatively affect well-being. And then exclusion levels will alter or will show as an altered pattern of connectivity in the brain. So what are the measurements? Initially, we are trying to make brain health assessment. Then also we want to study the perceived exclusion based on gender using the schedule of sexist events. We are going to evaluate psychological well-being and also the quality of life using the test that you can see here. Then what I think is a strong negative association between exclusion and well-being. This is not new, as you saw in the presentation this morning. We saw that people with depression has lower levels of independency and brain health. So it's something that we can anticipate, but we don't know how is particularly specifically for women. So more severe outcomes would be like observed in multiple discrimination environments or multiple discrimination conjunctions. And then the same for altered connectivity in women who live worse forms of discrimination. And these are like the whole references, well, not the whole references, but I have some of the references I've been working with. And I can say that particularly as we are going to use electroencephalography, I think what are we going to see is maybe altered power spectra, particularly alpha and beta band, as it's also being described in the literature. But again, it's described as people in conditions of any condition of vulnerability, but we don't have any description specifically for women. And that's why I'm interested specifically in brain health in women. Thank you. If anyone has questions, please have mercy. No, I'm sorry. Carlos, and then Dr. Jennifer, and then...
I'm sorry. Could you explain a little more about the hypothesis that you mentioned at the end?
Yeah, alpha band and beta band. Yes.
What would be the other variable that you are trying to correlate?
Like the way, like how the perceived exclusion in different levels, like for example, gender or ethnicity, are like being good predictors of these differences in the alpha and beta bands. Yeah. Carlos and Dr. Jennifer. Dr. Jennifer.
I just wanted to say, I think that this work is so fantastic because there hasn't been a lot of focus on women and aging and brain health for women specifically. So I think your work will be very important. I just had a question on the methodology. I wasn't sure if you said, is this going to be a study just comparing women with women in terms of their experiences, or are you also going to do a comparison with men to see if the sex-based discrimination has a larger impact on women's brain happening than men's?
We're working just with women, at least in these first stages. Thank you for your comments, Doctor.
I'm just curious about if you are thinking about how to handle the intersectionality that you mentioned about women. I'm just thinking here in Colombia about when you have, I don't know, maybe something about ethnicity, or you are part of a vulnerable population. How can you handle those different combinations?
We are going to use like this perceived, that maybe I don't know if it was in the title, but I am thinking about the perceived discrimination. It's like the higher scores that you put in your perceived discrimination is going to be are going to be put in the model to see if they have like any way of prediction. Sorry, I'm forgetting my English now. But that's it. Well, now we have our next presentation is going to be in charge of the person who just repented of participating, Cristian Avila. He is psychologist and he is our student of second year. And now my English is coming back. Thank you very much.
¿Cómo se presenta acá? No, ¿qué pasó? Sí.
Ah, sí. Good afternoon. ¿Sí? ¿Se escucha? No. Hola, hola. Hola. Sí. Good afternoon to everyone. My name is Cristian Avila. I am a doctoral student in the PhD program of neuroscience in Pontificia Universidad Javeriana. And my project the title of my project is Relationship between DNA methylation levels, social determinants of health, and brain health in a sample of individuals with dementia in Latin America. One of my main interests with this research is to better understand how inequality can affect biological mechanisms related, for example, to dementia and aging. I would like to begin, speak briefly about what epigenetic is. epigenetic is the study of the chain in expression of genes without altering the DNA structure of the DNA structure. We can imagine epigenetic as a set of instruction, biochemical instruction that tell to our genes how and when to work. There are different markers of epigenetics. We can see stone modification, neocoding RNA, and DNA methylation. Today, I will focus in DNA methylation. The DNA methylation is an epigenetic marker where a chemical group, a methyl group, is added in CpG Iceland, a set of bases that are found mostly in the promoter region of the gene. The promoter region of the gene are central in the regulation of the expression of the of the gene. When there are so many methyl groups in the promoter, the gene are silencing, are less expressed. But when there are less, as we can see in the image, when there are less methyl groups in the promoter, the gene is active and normal. When there are so many methyl groups, the gene are repressed or silencing. Methylation is a intrinsic biological process. Respond to biological internal signals. Nevertheless, has shown to be sensitive to external factors. External factors like environmental factors such as pollution, external factors like lifestyle factors such as diet, such as exercise, and sensitive to social factors such as poverty, healthcare access, and so on. So, a new approach of epigenetics called social epigenetics, exploring how that social factors can affect epigenetic and specific epigenetic mechanisms. The second big topic in this research is social determinant of health. Social determinants of health are the conditions in what people born, grow, live, work, and age. That social determinant of health can generate disease problem, health problems per se itself, but also can affect epigenetic modification, epigenetic markers, and that epigenetic markers can affect a health. For example, we can see Alzheimer's disease. Brain health is a dynamic process, the third key process in this research. Brain health is a dynamic process along the life that encompasses different mechanisms such as cognitive, motor, and emotional. Okay, why it's important to study epigenetics in Latin America? First, as Dr. Juliana said, we are underrepresented. In the red area, we can see the total of epigenetic studies are in the European ancestry. And the other colors area are the other ancestries, like Hispanics, and so on. So first, underrepresentation in the studies of epigenetics around the world. Second, we have a diversity in ancestry and high admixture in our genetics that it, as Juliana says, it's important to develop of neurodegenerative disorders. 3, the nature, frequency, and intensity of the social determinant of health in Latin America are different to the rest of the world. Also, in Latin America, the social determinant of health play a key role in the development of the demencia in un porcentaje más alto, in a high percentage. Gracias. Además, besides, we can see an important markers of epigenetics in different demencias like Alzheimer, Parkinson, Lewis, and frontotemporal dementia. As we can see, there are important markers. In 2 reviews performance for our group, we can see also that in Latin America, the studies of epigenetics in dementia are only 5 studies, and in social epigenetic are 16. So the objectives of my project is to determine the relationship between exposure to social determinant of health, brain health measures, and DNA methylations in a sample of Latin American individuals with dementia. And the other objectives que se desglosan de este. The sample used participants from the Red Lab project was described today detailedly. 3 groups of these persons: Alzheimer, frontotemporal dementia, and control. Red Lab follow a standardized protocol in where there are a tool to evaluate social determinants of health, and we use that instrument, that section of the protocol or Red Lab. As a preliminary results, we evaluate a tool called epigenetic clocks that use DNA methylation sounds and can predict biological age, different to chronological age. Biological age total and biological age by organ. In these first preliminary results, we can see as a dependent variable epigenetic clocks, as an independent variable ethnicity and sex first, orange and red, ethnicity and sex. The squared R are not so high, but when we make a model of regression with the 2 variables juntas together, we can see that the squared R are so high. If we put in a one model of regression all the social determinants of health evaluated in Red Lab protocol, we can see that the squared R are so high, 0.5. Also with epigenetic clocks. Thanks.
Thank you very much. Now we are having our next doctoral student, Juan Diego. He is a second year student and he's going to present his work in peace and brain. So welcome, Juan Diego.
Thank you, Laura. Thank you all. I think that you should be, or at least I think that you're sick of my voice, of hearing my voice, but I will be talking to you a bit more. Well, as Laura just presented me, I'm Juan Diego Rodriguez. I'm a second year student in the PhD in neuroscience at Pontificia Universidad Javeriana. And I'm really interested about the effects of intergroup hostility on brain and community health. And we must begin from what is intergroup hostility. Intergroup hostility is a stable pattern of opposition and antagonism toward others, mainly for their group affiliation, and combines cognitive biases, such as attribution or racial prejudice, affective states like anger or resentment toward toward the outgroup, and behavioral expressions like rejection or over and cover harm. It is necessary for these three components so we can talk about hostility, even though research has mainly focused on the first one and the cognitive one. Why study intergroup hostility? Because in today's world, we are facing a context with rising hate speech, hostile online interactions, and political polarization that create fertile ground for antagonism to escalate to violence. Being Colombia, one of the most dramatic ones, as you can see in the graph. I'm going to skip to the slides very forward because we have a time limit. Also, hostility has been related to several health outcomes. Among those has been associated to, in the physical plane, to coronary heart disease, cardiac mortality, metabolic syndrome, type 2 diabetes, systematic inflammation, greater hostilytic load, reduced serotonin churning function, and white matter changes tied to Alzheimer's. Psychologically, it predicts faster cognitive decline, telomere shortening, more anxiety, stress, confrontational or avoidant coping styles, pessimism, and unrealistic expectations toward others. That ultimately worsens those physical effects that I previously said. In the community level, greater hostility erodes social fabrics, fuels exclusion and conflict, weakens high-quality relationships, and limits access to resources, all of which decrements well-being and makes more possible that a conflict escalates into violence. To explore the relationship and how culture or social context may vary and may affect hostility, we are currently working in a study that is to build a hostility index using variables of the World Values Survey that encompasses more than 60 countries and has more than 97,000 data. And we are creating an index using some of the proxies that covers rejection, covers conductual harm, at least disposition to harm, and affective hostility. And we also create a well-being index based on self-perceived well-being that the data used. And our objective is to prove if that same relation that has been reported to in the literature also shows in this data set. Our preliminary findings shows that effectively intergroup hostility varies among countries. We can see countries like Iran, Turkey, Egypt, who shows more marked higher hostility. This was from the 2017 to 2022 dataset, so this will not reflect today's context. But it shows that hostility change towards the culture or the social context that you are studying. When we make linear regression analysis, controlling for demographics reveal a small but significant negative association between hostility and self-reported health. happiness and life satisfaction, as you can see in the below graphics. And the law association suggests that while hostility may not be the main cause for the negative effects or the negative well-being, it surely has a negative effect on health, and allows to see that it's possible to measure hostility through a large data set, and even through a proxy of hostility, we can see this relationship. The above map suggests that when you cross well-being with hostility, the relationship in different cultures is different. We have cultures with both higher well-being and higher hostility. We have countries with low hostility and low well-being, and we have the relation that I have been telling you about, the negative one. So this is one of the things that we are going to keep studying through machine learning models. So exactly why is this pattern so variable among different contexts? To talk about a little bit about why is that hostility seems to have such a negative impact on health, one of the most complete models that I have found to explain it represents a cynical pattern in which more hostility leads to elevated reactivity and stress responses, who in turn produce greater exposure to conflict, because if I'm all the time thinking that others will harm me, it's very possible that I harm them first. And that also leads to greater risk-taking and confrontational behaviors, and that in turn leads to more isolation and hostile biases. I am more alone because I fight with everyone, and I think that everyone wants to fight me, so this leads also to great hostility. So it creates a cycle in which hostility erodes and creates more hostility. Despite the evidence I have shown you so far, there's still some key limitations. Amongst them, no currently model fully integrates the cognitive, emotional, behavioral, and cognitive components of hostility. Even the frameworks that I have just showed you, it's promising, yet it still treats ideas, emotions, and actions separately. Most research draws on clinical and penitentiary samples, so it's difficult and hard to generalize those findings to the general or the broader population. Also, those results are fragmented across disparate conceptual approaches. So everyone has a different definition of hostility, or defines violence as hostility or aggression as hostility. So it's very difficult to keep track of what is and what it's not hostility, especially because those are very interlapping constructs, leaving us with an incoherent, undefined understanding that it's still need to understand about hostility. So we propose a comprehensive model connecting hostility to brain health, and exploring the mechanisms and identity factors and how that could modify that relationship. To address those limitations, we have found that brain health, it's a construct to have a unique opportunity to unify the possible multiple effects of hostility on health. As we have seen across these last 2 days, brain health treats health as a continuum, letting us to detect subtle declines or potential benefits of hostility. It also allows us to integrate cognitive, mental, functional autonomy, and social domains with an eco-bio psychosocial perspective that includes group identity and community factors. So we have the study. It is all this context that we propose, and we aim to understand how intergroup hostility impacts brain and community health among Colombians. With this remain objective, identifying underlying mechanisms and their differential impacts on brain health, point-to-point the specific impact on brain health domains of hostility in brain health domains, and examine EEG dynamics associated with hostile behavior. We will use a mixed sequential design in which we will use qualitative measures through semi-structured interviews and focus groups on the experience in which Colombians have through to intergroup hostility in daily life. And with that, we will contrast with models that currently exist, mostly developed in the UK. And with that, we will adjust the skills and tasks that we need for measure these constructs from a contextual perspective. For the M1, we will use scales, interviews, and behavior tags. We will test whether higher hostility correlates with stronger stress reactivity, elevated allostatic load, risk behaviors, and whether strong in-group identity identifies these links. We will use several I don't know, mechanisms, I don't know, to measure hostility, we'll use several ways of multiple methodologies, because hostility, as I told you, has broadly an uncoherent way of studying, so we want a very robust measure. In the second aim, we will focus on pair hostility measures with cognitive and social cognition tests, and mental health and well-being and social network indices, expecting poor scores as hostility rises. And finally, we will seek to study how hostility modulates the EEG signal at resting state in those individuals who tend to maintain a hostile behavior pattern versus those who doesn't. And we also want to see that measure in computational task scenarios that combine hostile and non-hostile stimuli. With this, we seek to compare a pattern reported by literature where higher hostility is associated with lower gamma and delta band coherence and a frontal asymmetry in beta bands. So the current status and next step, we are currently working on the WBS study that I presented, the preliminary results, and hope to advance across this semester. We will hope to make the interviews and adapting the instruments and launching main data collection to next year. And we hope that the final of next year, we have some results to show you the next time we see each other. Thank you for your time. These are my data if you want to make any questions. Does anyone have any questions? Thank you.
Thank you, Juan Diego. Ok. No te salvaste. Aquí hay uno, ¿no? Bueno. Sí.
Yes, the man in the back.
Thank you so much, Juan. I mean, I'm working with you in this this research pipeline, trying to establish connections between the notion of hostility or intergroup violence, and if it could be related with well-being, mental health, or brain health in that case. But I was wondering, because we saw some of your preliminary results with using the World Values Survey, and you have differences across country in the levels of what you call hostility.
Mm-hmm.
So I was wondering if you think that those indexes in terms of hostility could be, I mean, explained by cultural differences, or how the people reply or answer to that kind of questions. I mean, or do you are confident or convinced that, for instance, I don't know, Peru and Colombia have more hostility rates than, I don't know, Argentina, for instance? What do you think about that? It could be, I mean, impacted by cultural roots, or for language, or the approach of that kind of question, asking of regarding to those problems, which is very difficult to grasp, you know. Mm-hmm. In some ways, if you are, question it, if you are hostile against others, probably you say, no, I don't think so. So I don't know what you think and what you frame.
I think that one of the main difficulties working with large data sets, it's maybe the way that people ask the questions, or maybe the population that they choose. I think, and looking through the data, I think that at least the Colombian sample was very centrada en la ciudad. So it doesn't mark really the broad of the country. But as we see the whole world and see more than 60 countries, I think that those differences about how people ask the questions became less important, because it is a difference that is worldwide. And also the World Value Survey, it's a data set that has a lot of time and a lot of refinement. So it's a very powerful data set to study this.
Thanks for this interesting presentation. And I wondered if you could provide more details about how will you approach the concept of allostatic load, and if you're using the concept of allostatic overload, and how do you conceptualize it? And also, if you could make sort of a breakdown of the moderating factors, or I mean, like, as this is a obviously multifactorial process, I think that there will be some protective factors and predisposing factors and precipitating factors. So could you make a comment on that?
The first one about allostatic load, we are still deciding if we were going to use overload, but we are going to see some of the marks that stress leaves in the body. So we are going to use the hip waist ratio. We are going to use, if we can, cortisol cell level. So we want a very, I don't know, can I speak in Spanish? Queremos usar como algunos de las medidas que nos permitan ver ya el rastro que dejó una alta carga de estrés en el cuerpo. Entonces queremos ver ese impacto que ya tienen las personas que han tenido un patrón hostil a lo largo del tiempo. Now for the second question, if I remember well, it's a confounding factors and the broad.
Not necessarily confounding, because they could be moderators.
Yes.
Both in a protective or in a risky manner.
Of course. A construct like hostility, intergroup hostility, it's very difficult because when I make a mind map of all the possible constructs that are related, it's huge. I think most of the more important ones are how the group makes you feel. So if the groups make me feel safe, if it provides me of satisfaction, if the values of the group, that are very important ones, because if my groups provides value by stepping on other groups, that will make more possible that I have negative effects on hostility. But there are also good ones. For example, what if we are hostile to groups that are Nazi groups or constantly degrading others? So maybe in that way, that hostility can lead to a better health outcome. So yes, it is a very integrated, bueno, intricado, un mapa muy intricado, pero efectivamente hay varios de esos. Thank you.
Now we're going to continue with our Seed Grants awardees. And the next person in the presentation is Dr. Maria Marcela Velázquez Toledo. She's going to talk about epigenetic clocks and dementia risk in Latin America, integrating clinical, cognitive, and neuroimaging correlates in a pilot analysis within the Red Lab network. Welcome.
Okay, so good afternoon to everyone. I wasn't expecting to be the next in presenting, so okay. Good afternoon, and thank you everyone for staying here so late. I'm going to talk about this research project who was proposed with the collaboration of Dr. Hernando Santamaria and who was elected for funding through the D43 project, which is supported by the Fogarty International Center. And it is titled Epigenetic Clocks and Dementia Risk in Latin America, Integrating Clinical, Cognitive, and Neuroimaging Correlates in a Pilot Analysis within the Red Lab network. So I know that it sounds huge, but we are just exploring hypotheses that we could eventually investigate in this context. So it is well known by all of us that dementia in Latin America is really a significant public health challenge. And unfortunately, the number of cases is projected to triple by 2015 due to rapid changes in demography. And yet, the access to early diagnostic tools, for example, are still limited. And unfortunately, this is more evident for underserved populations. And also, our disparities in social and economic contexts influence disease risk and progression, making early detection even more important in this context. And as already been said, Salud in the Congress, underrepresented, we are really underrepresented in global research efforts of complex neurodegenerative disorders. So it is pretty urgent to attend this need in Latin America and in our country in particular. So I'm going to make just a quick mention of this topic because a lot has already been said about it throughout the Congress. But it is really important to mention and to remember the multidimensional and complex nature of the etiology of dementia, because it emerged as an interaction between biological or non-modifiable factors interacting with environmental exposures as well as social contexts. And a field that can lead us to emerge these different layers of risk factors is epigenetics. So Christian already gave us a pretty well context about epigenetics, but just to recap, epigenetics is the study of changes of processes that could change the level expression of different genes, and that is not related to changes in the DNA sequence itself. So what is amazing and fascinating about epigenetics is that it is a field that shows us that actually environmental exposures combining with genetics predispositions interacts and can lead us to make like a kind of biological interprints that can lead us to modify disease risk in different contexts. So specifically by the case of epigenetic clocks, we can think about it like a sort of chemical signature that not just reveal or reflects the passage of time, but also the changes of physiological processes as well as environmental exposures, for example. And for instance, this figure shows us the way that epigenetic clocks are developed and what they can tell us about the process of aging. So the starting point, it all begins with huge or large population samples from which DNA methylation profiles are estimated at individual levels, particularly at specific CPG sites, which are specific genomic regions that are liable to methylate it in response to different environmental conditions. So by providing information of the DNA methylation profiles of these different informative CPG regions, we can then use different statistical modeling, for example, general machine learning modeling, to choose that kind of the more informative CPG sites to then construct predictive algorithms to construct or to finally give us information about the epigenetic age. So the resulting epigenetic clocks, like for example, this one like Horvath or PhenoAge or GrimAge, ends up by not just showing us an idea of the chronological age, but it also could reflect the biological age and also processes like, for example, risks for disease and also mortality. And what is even more fascinating about epigenetic clocks is that we can use them to assess whether, for example, a person can aging faster or slower than expected. So for example, if the epigenetic age of an individual is higher than their chronological age, we are talking about acceleration of the DNA of the methylation age, or the epigenetic age. And it is constantly associated with a higher increase in the disease risk. And if it is lower, it is more an indicative of a healthier process of aging. So ultimately, these epigenetic clocks give us information or the combined effect of different molecular and biological processes. So it makes them really a valuable molecular, like kind of a outreach that is really worth to explore in the context of complex disorders like dementia. So with this project, we aim to address, first of all, the gaps that we have in Latin America to perform like a multifactorial or multidimensional analysis of dementia. So we are planning to leverage already existing data that has been released through the Red Lab network as a result of the huge effort of the different working groups within the network. For example, here in Latin America and in Colombia, led by the research group of Dr. Santa Maria, Dr. Matallana. So we are planning to leverage that huge amount of data to integrate data sets, and we want to explore both. A first scenario of a case control proxy to determine potential indicators of disease risk, but also to use, for example, dimensional analysis to explore possible or potential correlations between epigenetic acceleration age with different layers, variables, for example, cognitive, performance, or atrophy of different brain regions. So ultimately, this objective will lead us to propose like specific region indicators of dementia risk and progression for a Red Lab cohort of almost 800 individuals, including cases diagnosed with Alzheimer's disease, frontotemporal dementia, and also healthy controls, from which we have available these different layers of information: epigenetic clocks, clinical data, cognitive and neuropsychological measures, neuroimaging data, as well as social determinants of health. So this is obviously a part, for example, of the project that Christian is already performing, as you can imagine. And the second aim of this project is focused on capacity building, because we are aware that we have also really structural disparity challenges here in Latin America to promote research in dementia and other complex disorders. So we plan to strengthen this research environment here in our university. So we will include activities of tutorship and mentorship, and we are planning to involve a student for the PhD program in neuroscience here at the university to strengthen their skills in analyzing this kind of complex data. And it will also serve as an opportunity for me to grow as a mentor, as an early career researcher, which is my actual position here at the university. So this second aim is also a response to the need that we have to implement transdisciplinary approach to this kind of investigation in complex disorders. So we're hoping that through this project we can strengthen the academic collaboration between the Institute for Human Genetics, that is my institute, and with the Laboratory for Cognition, Neuroscience, and Context, directed by Dr. Santa Maria. And finally, the global expected impact of this project will lead us to propose like regional indicators of risk and progression of dementia in this particular cohort of the Red Lab network. And we are also planning to improve capacity building efforts, like making more available resources for both laboratories, and also by including promote knowledge dissemination activities, because we are really aware of the need by the social appropriation of knowledge. So we are planning to, for example, prepare some kind of community directed webinars in these topics. And ultimately, to make this more affordable and available for the general community, because it is a topic that here in the research environment is really excited, but we have the great challenge to translate it to the general population in order to see a real effect of that. So thank you very much for all the collaborators, and a specific acknowledgements for Dr. Hernando Santamaria for giving me the opportunity to propose with him this project. And so I was so serious about the 7 minutes, so thank you very much.
Thank you very much, Dr. Velasquez. I would like to know if there's a question in the public. It seems that we don't have questions. Thank you very much, Doctor. Oh, sorry, there's a question.
Thank you so much, Marcela, for your wonderful presentation. And I was wondering about because I'm aware that you have expertise in trying to assess candidate genes methylation for some neuropsychiatric disorders. If I understood well, you work with depression and other mental health conditions. And I would like to know your opinion about that kind of approach, epigenetics, and the potential impact that can have that kind of research pipeline in order to see the biology, environmental interactions that promotes mental disorders. I think it's also important, of course, for dementia and brain health. Not in doubt about that, but considering that the psychiatry conditions have that kind of interaction more explicitly, biology, environmental interactions are more clear, their role in psychiatry. I was wondering about it's important for me just to know your opinion about the potential impact that has this kind of approach.
Yes, that's pretty important. And as Christian mentioned, for example, there is a lack of research in epigenetics regarding complex neurodegenerative disorders. But for example, considering the relevance of social determinants of health, of the social context, of the even decisions in lifestyles that really impact brain health in general, and specifically biological processes relating to dementia and cognitive processes. So I really think that epigenetic is a really valuable tool to improve the knowledge about how can we intervene in some social contexts, and how we can track those changes and how they are actually affecting the biological mechanisms of gene expression. So yes, I find it really promising in this area.
Thanks, Marcela. I have a question related. So you said that you are going to have Alzheimer, frontotemporal dementia patients and the controls. How do you cluster all these people? Because maybe they have different social backgrounds, interactions, so that might affect your results regarding the methylation of these genes. I don't know.
Yes, that's a question that I'm still answering. But yes, we predict that there is a sample with high heterogeneity, and we are still wondering how to analyze this in the best way. But one approach could be to just cluster Alzheimer's disease patients and frontotemporal dementia patients as dementia patients and compare it to healthy controls. But I think that maybe a more valuable approach will be to try to wonder, for example, through dimensional analysis, analyze the whole spectrum of the sample like a continuum, and try to correlate, for example, epigenetic clocks with different kind of variables, for example, cognitive performance or shape of different brain areas. And it will be not an indicative of the risk of being diagnosed as a case with dementia or a control, but maybe just more accuracy, like a quantitative proxy of a correlation for example, linear regression models of dimensional models. But yes, this is a huge question that we still have to wonder to improve the results. Yes, thank you very much.
Thank you very much, Doctor.
Up next, we have Professor of the Faculty of Science, Dr. John Jairo Sutachan, who will be presenting neuroprotective effects of Lipia alba extract on neuronal and astrocytic plasticity in chemotherapy-related cognitive impairment.
So first of all, I want to thank you, Nano, for the invitation and also for the funding. It's a refreshing money that we really need right now. You know, funding has been so difficult in Colombia lately. So what we are going to try to do today, I'm going to try to do is just to give a long introduction of what we do, and then why this helps us to give the rationale of what we are going to plan to do with this project. So we have been working in the last years trying to understand how metabolites that are isolated from plants can regulate plasticity in neurons. And our focus in this grant is try to understand how this regulation may happen between the communication between neurons and astrocytes, and how this can improve plasticity, and then this plasticity can protect the brain against external stimuli that may lead to cognitive impairment. This is the idea that we have been basically working. And we have focused on cancer, and you will see how. Probably you know cancer is the second leading cause of death in the world. However, in the last 15 years, there is an increase in survival rates. It's about 20% increase. And this is mainly due to a better diagnosis, but also because there is a refinement in the treatments. And you can see here, for example, that happens in immunotherapy, also in radiation, chemotherapy, and bone marrow transplantation. However, because now patients live longer, we start seeing other side effects due to the treatments. And some of those side effects might affect, for example, the heart, or also they can affect the brain, and they can end up generating something that we call the chemio brain. So the chemio brain, we think, is something related with an acceleration in cognitive decline. As you can see in this plot, this is like an steady cognitive decline in normal aging. But then when patients receive, for example, chemotherapy, there is a suddenly decrease in this cognitive decline. And this has been well characterized in patients, for example, that has breast cancer and receive chemotherapy. And we can see here the numbers during the treatment. Around 60 to 75% of the women have some cognitive impairment. And then after the treatment, around 45% of the women will end up having a cognitive impairment that will be for the rest of their life. And this is affecting basically learning, concentrating, and making decisions that might be easy for us, and they might be so difficult for them. We don't know much about the mechanisms that are behind this cognitive impairment. However, looks like it's a multifactorial problem, and it might be involved inflammation. It might be involved also in reduction in neurogenesis and gliolinesis, and also some impairments in synaptic transmission. And something that we have been working is in the last one that might be related with some impairments or dysfunction in synaptic and functional plasticity, in which neurons lost the connection, and when they lost the connection, they have a reduction in the dendritic arborization of neurons, so affecting in this way the connection between neurons. And this is not uncommon. This is something that you can see in many other disorders. You can see, for example, in schizophrenia. You can see also in Alzheimer's disease, stress, and Down syndrome. And you can see that there is a decrease in the dendritic arborization and also in synaptic connection and basically into the spines. So in the last year, we have been working on understanding how this happened, but also to try to look for solutions to this problem. And we have focused in secondary metabolites isolated from plants that are called flavonoids. And flavonoids are quite important for plants because they can regulate growth, they can regulate development, but also they protect plants against some environmental factors like radiation. And so we start these studies evaluating, for example, some well-known flavonoid that is rutin, and rutin is a flavonoid that has 2 sugars. And when we treat the neurons, I'm going to use here, when we treat the neurons, you can see that there is an increase in the complexity of the dendritic arborization. They have longer dendrites, and you can see it here in this plot. When we try to figure out what might be the mechanisms behind this increase in the dendritic arborization, we found that rutin can increase the activity of the neurons. It's like having more action potentials, and those action potentials increase the activity in the neuron, and then looks like there is an activation of mechanisms related with synaptic plasticity, and looks like those mechanisms may involve in some way NMDA receptor on also AMPA receptors. It's like these are really good molecules to induce not only functional but also structural plasticity, as you can see here. However, Colombia is a biodiverse country, and you can see that in plants, together with Brazil, we are the countries that may have more plants. And usually those plants are rich in flavonoids. And this makes sense because we are in the tropic, and we have different altitudes, and we have different temperatures. So plants need to adapt to those conditions to survive. So we decided, considering this, to see if not only this kind of flavonoid like rutin, but also like a mix of flavonoids may have some effects in this plasticity. And we generate an extract from a plant that is Passiflora edulis. This is quite similar to the maracuja. And this is an endogenous plant from Colombia and South America. And you can see that when we treat the neurons with this extract, we also have this increase in dendritic arborization, as you can see here, and also here in the plot. And it's quite interesting because we also try another flavonoid that is called quercetin. And quercetin is like the father of rutin. So quercetin is the rutin without those sugars. And you can see that quercetin is not able to induce this plasticity. And this suggests in some way that not all flavonoids are able to induce structural and functional changes in neurons. And as I mentioned before, we focus in cancer, and we set up this system in the lab to study the chemiobrain. And what we do was to treat mice with doxorubicin, that is chemotherapeutic agent used in breast cancer. And we treat the animals with the doxo, and in some of these animals, we also treat with the extract from Passiflora edulis. And you can see what happened with doxo, and it's not strange that the people that are having chemotherapy may have some cognitive impairment during the treatment. That you can see here, this is a dendrite, and this is the control, and you can see that mainly the dendrites lost all the spines that are important for connections. However, as you can see here in the last panel, in this one, when we add the doxo plus the extract, we recover the spines that has been lost due to the doxo. So these results indicate this is not just an in vitro stuff, but it's also happening in vivo experiments. And we're wondering if we can translate this into changes in behavior. And as you can see here, we do these white maze experiments in which you can evaluate short-term memory. And you can see the animals that are treated with doxo, the red ones, have problems in short-term memory. However, when we treat these animals with the doxo plus the extract, the function is recovered. It's suggesting that they may use as a protective agents during chemotherapy. However, our model has a big caveat. We have been focused so much in neurons, and probably Gloria Patricia knows that the nervous system is more much than that. And also in this context, there are another cells that might be important for what these extracts are doing. And so now we are focusing a little bit in trying to understanding through this grant how the interaction between neurons and astrocytes might be affected for these extract-rich in flavonoids. And so we have these experiments. These are preliminary data. So what we do here is to treat just cultures, only neurons, and also we treat culture of astrocytes with the extract, and then try to use this extract to treat neurons and see if we have an improvement also in the plasticity. And you can see it's quite interesting that when we took the media from the astrocyte that has been treated with the extract rich in flavonoids, the extract now doesn't work, doesn't induce any plastic changes, suggesting that in some way the extract is modifying the profile of this astrocyte. However, when we culture astrocyte with neurons and they are in physical contact, we have the effect back in plasticity, suggesting that there is a connection or there is an interaction between astrocyte and neuron that is important for what these kind of extracts are doing. And also that happened when we treat these neurons with doxo. As you can see, the only way that we can protect these neurons and induce plasticity is in co-cultures in which we have the neurons and also the astrocytes. So now we are start paying more attention to interaction. There is not only astrocyte, but we have also microglia cells. We have oligodendrocyte that have been also shown to be important for plasticity. And this is what we want to do with this grant funded by the NIH Fogarty grant. So what we want to do now is go deeper in what is happening in the dendritic tree and also in spines and see if in some way we are regulating astrocytic activation due to the doxo. But also we want to move into more physiological experiments, and we want to see if in some way we are modifying connectivity, like making more synapses, right? And also we want to evaluate through this grant what might be the signaling pathways that are involved and how we can correlate the molecules that are present in this extract with the signaling pathways involved in these structural and functional changes. So I want to end it up giving thanks to all the people that have participated in the previous studies, all the people in our lab, also the collaboration with Andre Riveros in the Universidad del Rosario, Sionol King with the Colorado State University, and also with the people in the Instituto Nacional de Salud, Orlando Yaura, and all the funding that we got from Colombia Scientifica. So thank you.
Thank you very much, Dr. Sutachan. I would like to ask if...
Hola, John, ¿cómo estás?
Hola, Gloria, ¿cómo vas?
Muy bien, gracias. Qué gusto escucharte. Tengo dos preguntas. Una es en relación a la biodisponibilidad de los flavonoides en el sistema enterohepático.
Okay.
Es muy bajito, es del 1%.
Okay.
Entonces, en la proyección terapéutica, ¿han pensado de pronto en un futuro cómo sortear eso?
Sí, esa es una buena pregunta y la pregunta también que surge es qué es lo que llega al final al sistema nervioso central, porque se sabe que muchas de estas moléculas sufren modificaciones, ¿no? Hemos pensado de pronto trabajar con algunos nanoencapsulados, trabajar con gente con nanopartículas que nos ayuden a poner estos flavonoides y que ellos puedan pasar mucho más fácil, por ejemplo, la barrera y poder llegar en mayor cantidad al sistema nervioso. Entonces, esa es una idea que tenemos, que estamos considerando.
Piensa en la administración intranasal. Yo creo que eso tiene mucho potencial.
Sí, sí, eso es muy cierto.
Bueno, y la segunda es, o sea, estoy tratando de ponerme allá en el tejido cerebral y hay un problema de tumorogénico y de neurodegeneración tras la quimio, ¿cierto? Para bloquear la división celular del tumor. ¿Cómo crees tú que logrando pues pasar ese sistema enterohepático, llegar o vía intranasal, etcétera, que los flavonoides tienen una acción protectiva diversa, ¿cómo crees que entraría a sortear ese equilibrio entre una situación de proliferación versus una de deterioro propia por la quimio? O sea,
Sí, mira que esa es tal vez una de las preguntas más retadoras, porque muchos de estos extractos de flavonoides actúan activando vías de señalización que son importantes para la división del tumor, ¿sí? Sin embargo, hemos hecho experimentos in vitro con algunos de estos extractos donde hemos visto que no afectan el efecto del quimioterapéutico sobre las células, o sea, no impiden que el quimioterapéutico actúe sobre las células tumorales. Eso es bien interesante a pesar de que activan vías similares, pero sí es algo importante de considerar que aparentemente, por ejemplo, vías como las PI3 quinasas, MAP quinasas, que están regulando el crecimiento y la división celular, son las mismas que utilizan estos extractos para plasticidad, sí. Pero al menos los resultados preliminares que tenemos in vitro muestran que no afectan lo que el quimioterapéutico está haciendo en células que se dividen a un rate rápido de división. Pero eso es lo que tenemos en el momento. Gracias. Creo que había otra cuesta allí. Pregunta, Cristian, creo. ¿No? Ok. Vale.
Muchas gracias.
Muchas gracias.
Next, we have Dr. William Ricardo Rodriguez. He is presenting two projects. He's presenting exploring speech-based AI models and acoustical analysis for predicting cognitive decline in older adults and predicting mental health diagnosis in older adults using clinical scales and artificial intelligence. Welcome, Doctor.
Thank you very much. The first thing that I want to say is I am not from medicine or science. I am from engineering, faculty of engineering here in Javeriana. So maybe you detect some errors in my speech, but no problem. Okay, the first presentation is called Predicting Mental Health Diagnosis in Older Adults using Clinical Scales and Artificial Intelligence. So I want to start with this reflection, maybe related with how many technologies does a cardiologist have to do his or her work. If we think cardiologists find many technologies in different fields, aspects, applications. For instance, in monitoring aspects, a cardiologist find Holter technologies, portable technologies, artificial intelligence technologies. From therapy, they can find many options, or for instance, integrating different kinds of images, clinical images, or in rehabilitation aspect, many technology, diagnostics, AI-based tools, robotics in surgery, whatever. So for many fields in medicine, there are huge technologies. But about this reflection, but in the case of the psychiatrics, how many technologies they can find. I am talking about commercial technologies, final user technologies. If you have the money, you can find some device or technology. How many technologies find, for instance, a psychiatrist to do his or her work? In my experience working with Intellectus, they work with traditional clinical assessments, applying different scales. So in principle, there are a big lack in the application of technologies in this field of the medicine.
we know, and we observe in this Congress, other options, for instance, in the development of biomarkers, or maybe you can find some application of neurology, processing signals of the brain, or digital image processing, applying AI, artificial intelligence, to detect some specific responses. And engineers, we as engineers approach these phenomena to control other type of technologies or devices like robotics or similar like that. So let me explain the traditional Intellectus clinical assessment. This project was conceived with researchers from Intellectus. And the traditional way, from the patient at the beginning of the process to the end, the process is similar to this. In principle, a personal information here is not relevant. A patient start with the assessment and start with individual assessments from different specialties in Intellectus. You know that interact with geriatrician, physiatry, neurology, neuropsychology, and each of these specialties apply different scales, qualitative scales, quantitative scales, and obtain different results, values, and important information that after this process in interdisciplinary assessment in group, the professionals evaluate again the results and they create a final report, and they say, for instance, this patient have this diagnosis or this or whatever. This is the traditional way in Intellectus. So in this point, we find a point of intersection with engineering and with different algorithms or technologies. For instance, scales from geriatrics, psychiatry, neurology, and neuropsychology maybe in conjunction we have 200 values per patient. Suppose 50 patients per week. We have now more or less 10,000 variables or parameters for one patient, for 1 week. And for 4 months, for 12 months, for 7-10 years, we have a lot of information with results of these scales. So in this point, the question is, why not we use this information in order to train algorithms of artificial intelligence to support the diagnosis or support the analysis of this report. So with this part in mind, we propose the project. Our main goal is to evaluate the ability of an AI model trained with clinical and scale data to predict mental health disorders diagnosis in individuals at risk or with early symptoms, and its potential to support the effectiveness of the clinical assessment. The idea is, in conjunction with the traditional clinical assessment in Intellectus, apply these technologies in order to support or complement the diagnosis of the clinician. As a specific goal, we have, first of all, to adapt an anonymized clinical scale. For instance, we will ask the ethical approval to the committee to obtain the approval to collect the data, anonymize the data. After that, we want to develop and train AI models using this data. In this part, we want to use machine learning approach, so deterministic approach. For instance, apply random forest algorithm support virtual machines, Also, we want to explore deep learning with this information, maybe apply convolutional networks or similar. After that, we want to evaluate the predicted accuracy of the AI model in identifying potential diagnosis. For this, we want to do the test the system with new information from patients of Intellectus in order to compare the result from the artificial intelligence system with the traditional clinical diagnosis established in the Intellectus. Finally, we want to determine the utility and the impact of the AI model as a clinical decision support through the assessment of the mental health professionals. The idea is that the final clinicians assess the tools with different instruments that will be developed for that intentions. So this is again the root of the Intellectus assessment. In a previous slide, I explained a little bit of this. The change is, or the proposal is here. We want to obtain the parameters from these scales, from the 4 areas. With these informations and parameters, we want to train machine learning models in order to the system propose the probability to obtain this or that diagnosis and complement the traditional way in Intellectus to assess a specific individual. So for this flow of information, first of all, we wanted to collect the information, develop and validate the machine learning models. Here we want to apply, for instance, decision trees or random forest, supervised machines, or as I explained, others deep learning approaches to train the machine learning models. After that, the principal investigator and co-investigators, we went to analyze what are the main clinical predictors, or for instance, possible clinical developments or any other informations in order to complement or support the clinical or diagnosis proposed by the interprofessional assessments. So we want to expand results like, for instance, expand research or mental health problems here in Colombia, in our community, in our elders, obtain an initial platform to support the diagnosis from mental health, and to have the opportunity to try mental health problems at early stages. All of this is possible for, obviously, I hope I believe in the very happy marriages between engineering and doctors and medicals and science in order to obtain very useful and the application of the technologies with a real purpose. In this case, apply the artificial intelligence technologies in order to support the decision. I think there are a very huge lack of application of the technologies in special artificial intelligence developments and approach. We need to apply more algorithms, more technologies in our real cases, in our community, in our problems. So this is the first part of the presentation. If you have questions, I am here. Thank you.
Hi, thank you for the presentation and the work that you're doing. I'm curious, so I'm a social scientist, but we've started using machine learning, like regression trained neural networks, the XGBoost and all of these other kinds of machine learning techniques. But sometimes for us, it's difficult because it creates a black box and we can't really see what's happened, only what came out on the other side. With the approach that you're proposing, will you then be able to show a decision tree so that other people could look at it and see an algorithmic classification approach that could be used without always having to train a new machine learning model on every data set? Or is this really specific to the inputs of a specific data set and you would have to use another machine learning, another model that you trained on a different data set for that one? I guess my question is about transportability.
Okay, thank you. I understand. In principle, we want to reproduce some experimental setup from literature, and it's more common to use machine learning algorithms because you can explain with more arguments the predictors or the finding of the technology. Some cases when the researchers applied learning approaches, for instance, if you say to a doctor, for instance, the diagnosis is this, and I apply a deep learning approach, I don't have the interpretability of the result of the deep learning model. So it's so difficult that the medical professionals use one result of the technology if you can explain this result is from Y aspect, Y predictors. So the first stage of the technology, we want to apply a deterministic approach, a random forest or other approaches. Support vector machine is a very important predictors in dementia, for instance. For that reason, in deep learning, the interpretability of the model is difficult, and we need to continue with the research. For this part, we want to the machine learning or deterministic approach. Thank you.
Thank you. Any other questions? Perfect. I think we can move on to the next presentation.
Okay. We are going to continue with the second project. From this, I want to say that I am a biomedical engineer, and my PhD was in the processing of the speech signal from children with speech disorders. So the acoustical analysis of the speech signal is very important for me and my previous research. So with the opportunity to work with Intellectus, we detect one opportunity to apply or obtain additional information from the Intellectus recording the speech from patients. The speech has a very important information about the health, about the cognition of the patients. So for that reason, I propose with my colleagues this project called Exploring Speech-Based Models and Acoustical Analysis for Predicting Cognitive Decline in Older Adults. For this part, a brief introduction. We know cognitive decline in older adults represents a significant health problem for the patient, for the family, for the system, the public health system. On the other hand, as the global population there is an human need for application or innovation approach to support or to detect in early stage the problems, cognitive problems in that population. In this image, we can see it, for instance, our community, our elders, little by little, they need more specific and more attention and more technologies, and these situations pressure in a huge mode the medical assistant, medical doctors, technologies, and whatever. Also, as you know, for instance, in Japan solves more older adult diapers than baby diapers. Let me explain this part in Spanish. Japón ya vende más pañales de adulto mayor que de bebés. Era lo que quería decir en inglés. Lo que representa un serio problema. So with this information in mind, we definitely need to do and to develop technologies with real application in our society. In this case, we can find many researches and works related with the use of speech signals in elders. For instance, prediction of Alzheimer's disease progression within 6 years using speech, a novel approach with language models. In this case, very interesting research, automatic assessment of loneliness in adult artists using speech, analysis of response of daily life questions. This research began in pandemic for COVID-19. Our older adults experiments episode of loneliness affecting their wellness. In other examples, measurement of neuropsychiatric symptoms in the older adults with mild cognitive impairments based on speech and facial recognition, expression recognition. In this kind of technologies, the engineers and clinicians joined speech signal processing, image digital processing with facial expression, also apply different scales like in previous project, in a cross-sectional observational studies. And in these examples, that which is very, very similar and related with this project, automatic detection of cognitive impairments through acoustic analysis of speech. But please pay special attention to the corporations, Panasonic Corporation. Next, Department of Preventive Gerontology, Center of Gerontology and Social Science, etc. This research was made by corporations like Panasonic, nor like Osaka University or academic. This is a very interesting case that the companies are developing technologies for attending the elders. Here in Colombia, we are approximate to this situation similar to the Japan and other countries in where we need develop technologies for our adults. In this case, the acoustical analysis includes, for instance, analysis of the fundamental frequency of the voice, the formant resonance or formants from the vocal tract, also include prosodic information, intonation, time of speech, and different information in order to apply AI, and with this detect some specific or predict some specific cognitive decline in older adults. So in this case, the objectives of the project, our main goal is to develop and validate that speech-based AI model and acoustical analysis techniques to predict possible cognitive decline in older adults. As you know, cognitive decline may include many diagnoses, Alzheimer, dementia, etc. As a specific goals, collect scientific data from speech research databases. I will explain that in next slide. And extract acoustical parameters reporting the scientific literature. Second, to develop and train an AI model using the speech data collected. And finally, to evaluate the effectiveness of the speech-based AI model, comparing its performance with new data from Intellectus patients. So what is the idea? As I said, I hope, I believe in the happy marriage between engineers and doctors. So this union from Intellectus and speech from patients and artificial intelligence, I hope that this produce very good results. Similar, the method similar to the previous project, I said something about this route. The difference with this project is the patient that signed the informed consent, we obtain the speech signal, and here we obtain the acoustical parameters. But previously to this step, we need to train the machine learning models. But how? Fortunately, we can find on the web different research databases with information like, for instance, dementia, like Alzheimer, and other diseases with data like audio, video, transcriptions. So, for instance, Dementia Bank have a Dementia Bank Spanish Ivanova corpus from Spain. Also, we can find a corpus with English speech signals from patients with dementia, with Alzheimer, with normal, with health controls. And the idea is from this database and apply traditional algorithms for speech processing, obtain the acoustical and prosodic parameters. I said acoustical, for instance, the pitch frequency from the fundamental frequency on the vocal cords, formal frequencies, also capsule frequency, jitter, shimmer information about the variability of the fundamental frequency. In temporal domain, we can detect or calculate, for instance, delays in the speech. So similar in case of dementia or Alzheimer. Also, we can detect through algorithms number of pulses, number of verbs of different techniques for natural language processes. With these informations, we train the AI models, in principle, machine learning models. And with the trained models, here comes the signal from the new patients, and the system, as a result, say the probability of the cognitive decline is that number, or the probability to Alzheimer or dementia or similar like that is a number or probability in percentage, etc. Expected results also to expand research in mental health problems in all areas in Colombia, to obtain the probability of cognitive decline from speech signals, and to have the opportunity to try cognitive decline problems at early stage. Other aim that we have, other objective is the possibility to create our own database in Colombian Spanish, because there are differences between European Spanish and Colombian Spanish and Mexican Spanish and Argentinian, and some differences are detected in the prosody and acoustical parameters. So in principle, there are differences between the results between European Spanish and Colombian Spanish. So this is an excellent opportunity to create our own database in Spanish with cognitive decline or dementia or Alzheimer problems in speech of our community. And the idea is also share this information to research with other latitudes. Okay, this is the second project. Thank you.
Thank you, Doctor. I would like to ask you.
Buenas tardes, muchas gracias. Me parece increíble esto que estás haciendo como fonoaudióloga. Creo que es el sueño de toda fonoaudióloga tener estas bases de datos. Tengo varias preguntas, creo que muchas después te las haré, pero para este escenario en específico, ¿ya saben qué parámetros van a evaluar o solo son parámetros vocales, eso que decías de shimmer, jitter y frecuencia fundamental, o van a evaluar otros subprocesos del habla adicional a la fonación? Es la primera pregunta. ¿Qué quieres?
Sí, de una vez. Sí, en principio vamos a reproducir lo que sugiere la literatura y lo que he podido hacer en otras investigaciones. En la caracterización acústica definitivamente es muy relevante parámetros de tiempo y frecuencia de la frecuencia fundamental y algunos de los formantes. Adicional a esto, análisis prosódicos sobre el número de pausas, las latencias, la repetibilidad de algunas palabras, y variaciones prosódicas. Hasta ella hay suficiente información para alimentar los algoritmos y también utilizar subrutinas ya establecidas para manejar variaciones en tiempo y frecuencia de la frecuencia fundamental, jitter, shimmer y demás. Hay aproximadamente hasta 6.000 parámetros que yo pudiera obtener solo de parámetros acústicos. Viene el otro gran mundo, y te entiendo la pregunta, he trabajado bastante con fonoaudiólogos y viene muy al lugar. Esto es solo lo acústico, lo que yo escucho. Pero ¿y qué pasa con todo lo semántico y todo lo que yo quiero transmitir? El mensaje como tal de la comunicación es otro mundo grandísimo y veo que es como el siguiente paso, o si lo podemos hacer en conjunto, fabuloso. Vi ayer un póster muy interesante de Milena, en donde están trabajando la semántica y aplican modelos de lenguaje para detectar ciertas cosas en ciertas condiciones cognitivas. Sería ideal combinarlo con esto, pero entonces habría que articular los modelos de lenguaje, perdón, los modelos acústicos con los modelos prosódicos más los modelos de lenguaje y ver qué sale. Esta es la gran ventaja como son algoritmos, uno puede meterle cosas a ver qué sale, pero sabiendo siempre cómo interpretar las cosas con Machine Learning. Así que sí, en principio solo lo acústico, lo que mencioné y lo prosódico, pero todo lo del lenguaje, y eso es bienvenido.
Perfecto. Y en este banco que mencionabas que ya hay de demencias, ¿qué tareas de habla encuentra uno ahí?
Vale, hay varias muy interesantes. He pensado en reproducir dos. Hay una que está estandarizada y es, si recuerdo bien, la historia de una galleta en una imagen. Hay que describir lo que pasa con la galletica y eso genera ya un discurso fonológicamente balanceado en teoría y tiene ya algunas consideraciones acústicas y semánticas en esa tarea. El otro es un paper muy interesante, de hecho es de este trabajo de Dementia by Spanish Ivanova Corpus, fue hecho en la Universidad de Granada en España, tiene más o menos 300 registros de audio de diferentes condiciones cognitivas, entre ellas deterioro cognitivo moderado, Alzheimer y controles sanos. Y lo que hicieron fue colocar como tarea que todas las personas que donaron su voz para la investigación, incluidos los pacientes, los pusieron a leer los 2 primeros párrafos de Don Quijote, que ya per se tiene mucha riqueza, bueno, auditiva, fonológica y demás. Aquí pudiéramos pensar en algo así, ¿por qué no coger algo de 100 años de soledad y empezar con nuestras palabras y con nuestra manera de pronunciar, empezar a detectar esos rasgos acústicos, prosódicos, semánticos y demás, y nutrir nuestros propios modelos y compartir y demás. Vale, gracias. Su merced, creo que tenía...
No, te quería preguntar más en relación, primero, felicitarte por el trabajo, se ve muy, muy interesante. Es más desde la parte procedimental del análisis, en relación a la cantidad de datos, o sea, de casos que vas a utilizar. Hablo del propio banco colombiano. ¿Cuál es la expectativa? ¿Qué cantidad de casos quieres registrar que estén leyendo, digamos, 100 años de soledad, un par de párrafos? ¿Y cómo los utilizarías de referente en el Machine Learning para discriminar con los diferentes scores que tienes allí como indicadores de alteración?
Sí, muy, muy interesante pregunta. Efectivamente, en principio, con respecto a la primera parte, la expectativa es, por supuesto, dependemos primero que el comité de ética nos dé el aval, y estoy en esas. Lo segundo, que la persona que entra como paciente acepte, consienta que quiere donar su voz para la investigación, eso ya es una posibilidad. Asumiendo que la mayoría puede, hemos hecho estimaciones de si en Intellectus tenemos aproximadamente 50 pacientes en clínica de memoria, ojalá podamos obtener 40, 50 grabaciones por semana. Eso podría significar 200 al mes y fácilmente pudiéramos llegar, ojalá, a unas 2.000 por año. Si podemos grabar desde ya en año y medio, pudiéramos tener 3.000, no está nada mal. La base de datos de este demencia de España tenía 328. Ahora bien, lo primero que hay que hacer es que estos pacientes entran, la ruta habitual de Intellectus los clasifican sus diagnósticos y esas van a ser nuestras etiquetas para poder decir los parámetros acústicos para demencia son estos, los de deterioro cognitivo son estos y demás. Nos falta la parte importante de los controles. Ahí tenemos también que en la iniciativa del proyecto está hacer una grabación también de sujetos de las mismas condiciones pero sanos, es decir, que no tengan la condición cognitiva y poder tener como ese control. Después aplicarlo y empezar a hacer las comparaciones.
Porque yo también pienso que ahí se tendría que tener en cuenta que esté pareado por edad y sexo, porque influyen como factores, aunque estén dentro del rango de los casos sanos, que son referente interno para todas estas otras valoraciones, porque
Sí.
las mujeres en posmenopausia tienen cambios importantes en la fluidez verbal sin necesariamente, digamos, estar en fase de deterioro cognitivo. Entonces, eso, como...
Sí,
tener en cuenta esas
efectivamente.
variables.
De hecho, encontré otras investigaciones, de las primeras que mostré, hay estudios con 12 y 18 pacientes, porque la clasificación y el pareamiento final fue tan difícil que solo lograban unos cuantos de control y del el diagnóstico. Es muy difícil. Si tenemos al final 2.000, si llegamos a 20 y 20, es ya un gran éxito. Entonces hay que considerarlo. Y otra cosa que puede afectar mucho los resultados aquí es la escolaridad. Yo puedo tener dos perfiles idénticos, pero con el cambio de escolaridad, si uno es de 4 o 5 años de primaria y el otro tiene 15, 20 años de escolaridad con doctorado claramente, y eso se ve mucho en la semántica, los resultados son otros. Entonces, el pareamiento es difícil. En principio, entre más datos, pues mejor. Yo pienso que este y el anterior proyecto son excelentes oportunidades para empezar a trabajar en esto, porque tenemos los pacientes, tenemos la voluntad, tenemos los profesionales, pues ahí es que empezar a trabajar. Gracias.
Gracias, doctor Rodríguez. Well, thank you all for staying with us. Thank you, Dr. Rodriguez. I have good news and even better news. The good news is that we are still going to have some time together. And the even better news is that the person we are being waiting for has finally arrived, Dr. Curley. Please receive Dr. Curley. Dr. Curley received his undergraduate degree from the UCSD and a master's and PhD in behavioral neuroscience from the University of Hawaii and completed a postdoctoral training at the John A. Burns School of Medicine in Epigenetics, Immunology, and Infectious Disease. He is currently Associate Professor of the Department of Medicine, the Division of Geriatrics, Genetology, and Palliative Care at the Sam and Rose Stein Institute for Research on Aging and the Center of Healthy Aging. Please, Dr. Corley. Of course. Let me. Is this one?
That's my oldest daughter. Okay. So first, thank you everyone for staying. I just came from the airport, so I spent 10 hours in Panama. I got caught in the clouds this morning, so I'm really excited to be here. And today I kind of wanted to show you a little bit about where we're going in the future and stimulate some conversation about epigenetics and the environment, a new horizon in neuropsychiatry. So the goal of our research is to study how to extend the portion of life spent disease-free. So we really like to think about brain health and brain disease in terms of the aging process. And for us, I think in terms of looking at disease onset and different things people are interested in studying, this process, I think, is really related to understanding the human lifespan, but more importantly, the human health span. So the number of years you spend living without disease. And for us, this has really been a large focus. And this is my most recent appointment, moving to a division of geriatrics and working in the aging field now, allows me to both use my neuroscience expertise, but also, start to think about other things in terms of other parts of the body and other pieces of the environment that influence brain health. The brain itself, isn't going to be an isolated kind of component. We need to really consider a holistic view and capture as much data as we can. So really, I think this gets to the fundamental question when you look in the literature and across different scientific domains, and this has been a question posed by many different groups, and a real, I think, fundamental foundation to think about things. What matters more for our aging trajectory? Is it your genes, your lifestyle, or your environment? For me personally, in our research program, we think all of these components are critical to the aging process and to healthy aging. This field here is, I think, really starting to evolve in terms of thinking about how we disaggregate the time someone spends chronologically. So just think of that in terms of the years spent living, in terms of a year passes, so you're a year older, and adding on to that concept, the idea of biological age. So the difference essentially between what your body is aging at versus the concept of just chronological age, which is a continuous variable over time. And we really study the interplay of how our genome, so the DNA and environment, impacts our aging trajectory. And for us, this really is important to start to get experts across different domains to understand the role of the environment, exposures, the exposome. Right now, pollution is being, I think an area that a lot of studies are looking at in terms of microplastics, and environmental things that are accumulating in people's body and how this potentially could impact biology, poor diet, social stressors. So, this mental health aspect that really, I think, sets up the stage for the aging process and how brain health will proceed. And the other emerging concept, which we're well aware with in terms of looking at infections. So, in terms of potential things in the environment that are leading to impacts on health longitudinally. In terms of this field, I put this slide up here just to highlight for those that aren't familiar with this field. It's a field that really is merging multiple different domains of research across medicine. The field of geroscience. So this is a field that studies genetic, molecular, and cellular mechanisms that make aging a major risk factor and driver of chronic conditions and diseases of older people. So this field is really interested in studying the underlying mechanisms that people think are leading to disease. So for us, the hypothesis is essentially that a large component of disease is driven by this aging process. So if we can start to disaggregate these mechanisms, we can really start to think about interventions or recommendations to impact healthy aging and to hold off kind of that disease onset from occurring at an early stage, so you can increase the number of years you spend with a good health span. And really the hypothesis in the geroscience field, what their goal is, and really what our goal is to slow the aging process, to improve health and delay the onset of chronic disease. I don't consider myself a person that gets into the aging field in terms of looking at immortality. The clock is going to tick continuously. We're not going to stop or reverse the clock or basically, live forever. But what we could do are make these changes in terms of how quick we are aging, and age at a rate that we should be aging for our age. And, we think that this potentially could have huge impacts in terms of the onset of chronic disease. When we start to think of geroscience hypothesis and brain health, it really makes sense. So in terms of if we're plotting the chronological age on the x-axis here and the incidence of comorbidities, the geroscience hypothesis essentially is saying that comorbidities are increasing, which the geriatrics field knows across time. And when you start to look at brain health and deterioration in terms of mental health and other aspects as well, the geroscience hypothesis is basically stating that if we can start to target some of these fundamental processes, we essentially can improve brain health or essentially make that process age at the rate the brain should be aging. In terms of what we're studying, we're really looking at how the environment interplays with our biology. Different groups kind of sit at different levels here that I'm presenting, but fundamentally, I think all of them are important. The most well-studied has been genetics, so our genome, every cell in the body, essentially has, for the most part, besides these new emerging somatic mutations, the same DNA sequence. So that DNA sequence is kind of the recipe book that sets the stage for how cells will respond, but more importantly, how the aging process proceeds throughout time based on some potential genes related to aging or healthy aging that may be protective. Our lab has really been focused on the epigenome. So these are these chemical marks on top of your DNA code that are essentially thought of as you were a musical conductor, you read a musical score, but that piece of music is played differently every time. And this is the message that we're studying, these covalent modifications. And this information as well is giving us insights in terms of aging. So this is this epigenetic clock idea that's emerged in the field. And really all that is taking machine learning, applying it to data where we know someone's age, and then we're able to tell you which of these marks are important for measuring someone's age. You have the transcriptome below that, which is essentially RNA that's produced. This is a more dynamic process, proteins, and then the metabolome, which are different metabolites in the body. So our lab, as I said, is really studying the symphony in your cells, the field of epigenetics. This here is probably the best analogy for me I give to people that aren't, experts in the field of epigenetics. Your DNA is that piece of music. The epigenetics are that addition on top of that musical score. So these chemical modifications that essentially you could erase or put down in pen. And the phenotype gives rise in terms of, what we're seeing in terms of biology or aging processes, or more importantly, brain health in terms of how the brain functions. And in totality, you really can start to do this across the body and across different cells. So essentially the musical score that's played from all the cells within our body, we essentially are looking at what that conductor is doing in an individual and reading out what that musical score should look like from each person. The other analogy that's in the field, a little bit more simple analogy, is we're really looking at where these potential light switch or on-off signals in terms of different messages that are there in terms of which genes could be functioning on or off. In terms of aging, as I said, the field has really emerged recently and really done a good job, I think, in terms of identifying the hallmarks of aging and age-related disease. So what this slide is showing you here on the left, these are the hallmarks of aging. So these are the things that we think that are underlying the aging process that are leading to cellular changes that are essentially driving that aging process. These could include a dysbiosis of someone's gut, chronic inflammation, you have genome instability. Telomere length is another thing that's been studied quite well. As I said, epigenetics, mitochondrial function. And then you also have this emerging idea of stem cell plasticity, so how much your cells are able to respond as we get older. The amount of stem cells you have decreases over time. And then on the right-hand side, you essentially can take a lot of these common age-related diseases and look and see that the drivers of these diseases are related to these hallmarks of aging. So really, this is the kind of idea of geroscience. If we really can understand, okay, mitochondrial function is impaired, how could we maybe improve mitochondrial function? This may potentially hold off the onset of a disease that's related to a loss of mitochondrial function or vice versa. So this is really trying to piece together the overlay of age-related disease and these hallmarks of aging. In terms of our lab, we heard some great talks today. I saw on the schedule looking at epigenetics and the marks. These marks that I referred to in terms of that conductor, looking at these pencil or pen marks, are DNA methylation. These are chemical modifications. So the interesting thing here is we can get a DNA sample essentially hundreds of years old, and because it's a covalent modification, it's very stable, and we can measure what that methylation pattern is on someone's DNA. And this allows us to develop epigenetic clock, as I said. And I think this was probably discussed, but just very briefly, these clocks are really just multivariate predictors of biological age and lifespan. So if I take 1,000 people in a room and I know their actual age, and let's say we have people ranging from birth all the way to centenarians that are 100, and then we have methylation patterns from every single person in the room, we take those 1,000 samples, and then we ask the question, which methylation marks are predictive of those people's chronological age? And what we find is that we get a very highly accurate predictor. So we can take this data set and just measure the methylation marks and apply it to anybody, and we essentially can tell you what these individual's age is. And this has really led to kind of this emergence of these Horvath clocks, Hannum clocks. We've built our own clock looking at retroviruses. But simplistically, all it really
boils down to is having a very good epigenetic data set and good metadata, and then training a clock and showing that as predictive validity. And really right now, the field is kind of progressing very quickly. We have a couple projects currently to try to update some of these biological predictors, because you can imagine the data is only as good and it only works in populations where the data came from. Not surprising for people that are in that kind of data science or machine learning space. The more data we have, or the broader the data set is, the better the generalizability. And we can start to use this data as well in terms of looking at, okay, well, now we have a way to tell holistically what someone's age is and whether there's an onset of disease or if they're aging quicker or slower. And this has really been kind of this preponderance of a lot of papers in the field showing that there's factors that are associated with if the clock is going quicker or the clock is going slower. Certain diseases are associated with this particular measure. So we really think that this is a potential good biomarker to be using in terms of the aging space, but also for brain health in terms of looking at how this may interplay with the brain. And to do that, we took a recent project which was just accepted for publication, working with a very talented radiology fellow at UCSF. And what we did was looked at data in the Alzheimer's Disease Neuroimaging Initiative, ADNI. And we kind of had this idea. I'm coming from the epigenetic space, but I do have a very good neuroscience background. And we had the idea, okay, well, maybe we could take some of the ADNI brain health measure data, the imaging, and could we kind of look at the relationship between someone's aging rate with these clocks and some of these brain-related measures in terms of whether there's atrophy or progression. So are people progressing to AD quicker, or is their brain actually being measured in a way that's proxying biological age? And what we found was that DNA methylation age from peripheral blood predicted the progression to Alzheimer's disease, white matter disease burden that Luke was able to quantify from ADNI, and cortical atrophy. In terms of the cohort, I'm just showing the table there that Luke was able to harness the data set from. And in this particular project, there were 121 individuals that had longitudinal data with DNA methylation data paired with brain imaging data, and as well as all the Alzheimer's biomarker data. There were 236 participants that were classified with MCI. And then we had 46 participants with Alzheimer's disease. For those that are familiar with ADNI, you're well aware, but those that are not, ADNI was trying to look at progression to AD, so it wasn't a cohort that was enriched for AD participants. This is just showing you what the data is looking at. These are survival plots stratified by epigenetic age. So we looked at individuals that were in the 90th percentile and the 10th percentile. So essentially those participants that were aging quicker or aging slower, and that who were cognitively normal at baseline. So here we're looking at the progression. So these people came in, they had an epigenetic test, and their test essentially gave us their aging rate, but just based on the actual cognitive score, they were cognitively normal. And what we found was that two particular clocks, so this one DNA methylation phenoage, it's significantly associated with progression to MCI or AD. And also there was a trend in another clock, a mortality predictor that also trended in the same direction. So essentially if you were aging quicker, you progressed to MCI or AD at a higher likelihood than those individuals that were aging slower based on these epigenetic clocks. And then we also looked at Cox proportional hazards models where we covariate for sex, education, CDR, SB score, and ApoE 4 dose. And we looked to see whether that had an impact, and it still seemed there was a signal there driving from the biological age process. So this was an additional add-on potentially to things that people had shown to be influencing progression. So for us, it kind of gives us this interesting insight just at a baseline level, taking some data, looking at cognition and pairing that with some of these epigenetic biomarkers, showing that they could be potentially predictive and used and improved upon, I think, as well, in terms of looking at progression in people over time. In this particular context here, it's looking over 10 years. And then these are regression plots in participants that were controls and mild cognitive impairment. And then we're stratifying based on epigenetic age within, so -1 standard deviation corresponding to the 16th percentile, +1 standard deviation, which is the 84th percentile. And then the mean line there is presented in that dotted green line.
And what we found that epigenetic age predicted cognitive and clinical changes during normal aging and early neurodegenerative disease as well. So this was looking at the relationship between epigenetic age and worse clinical scores as measured by CDR-SB and MMSE scores. So this was taking a little bit of extra data and doing the same thing, but looking at the relationship here in terms of the aging rates of individuals. And then the next thing we did, which I thought was kind of the more exciting thing, and this is something we're excited to work with others on, is take some of the epigenetic age data and really integrate this with people that have a lot of neuroimaging expertise. There's been a lot of beautiful work, both I think here in the group and from others, looking at taking neuroimaging and calculating a brain age from the neuroimaging data. In this particular context, we looked at whether this peripheral biological age marker of epigenetic age related to the actual brain measures. So for us, I think this has a potential too to be utilized in the future, where maybe there's a particular area that we're not able to get a brain image on someone. You essentially could do a blood test and proxy someone's brain health. That's kind of the general idea here. So it's really scalable at that level. And we found that epigenetic age predicted cortical thickness across the spectrum of healthy aging to neurodegenerative disease. In this particular analysis, that was just repurposing free surfer data from the ADNI data set. And then we also looked at, again, correcting for multiple regression models where we corrected for sex, education, CDR-SB score, ApoE, E4 dose, and then for those that are brain imaging experts, total intracranial volume as well. And that relationship still held up, showing potentially that these aging clocks are capturing brain health measures relevant to neurodegenerative disease. And this has kind of stimulated an idea of looking at these brain clocks in the periphery, which could really be trained on having the neuroimaging data together in one kind of composite measure. So the more data you have with the epigenetic clocks, you really can utilize the data to develop a very sensitive test. And there's been thoughts too to include potentially the Alzheimer's markers. So if we have p-tau levels, if you put that in with the epigenetic markers, it'll become more sensitive to really pull out certain phenotypes that are of interest. And then this is just showing you data of epigenetic age predicting cortical thickness in healthy controls as well. So it seems to be not an effect just driven by disease in the sense of, okay, individuals have very significant differences in their brain imaging modalities. It seems to also capture smaller differences across individuals that seem to not have overt neurodegenerative disease as well. So just to summarize, in terms of the existing literature, epigenetic clocks are informative markers for multimodal measures of brain health. Here I'm just highlighting other groups and papers where people are starting to think about similar ideas. And essentially what it's taking advantage of is the incredible neuroscience and neurology-related measures in people and sub-phenotyping them, and then incorporating these aging markers and really starting to think of, okay, well, this is an additional variable that has a large impact in terms of brain health and how we think of the aging process, and more importantly, I think related to neurodegenerative disease, different features in terms of progression and other aspects that we want to look at in terms of treatment. Because currently, I think in terms of the Alzheimer's space, there's going to be a lot of interest in terms of doing personalized medicine. So potentially having someone's aging rates or aging biomarkers will really also help inform kind of, what interventions may be suitable to people that have that particular phenotype to maybe harmonize with some other aspects of, removing potentially the things that are in the brain causing the issues. So just to summarize, today I just wanted to keep it brief, but I think, the things that all of us know are the most fundamental to brain health. I'm coming into the aging field in that space, not trying to sell you some sort of supplement or some drink that's going to make you age at a better rate, but really the best data we have and the most rigorous data I can tell you is healthy habits. As we all know, good diet, exercise, and stress reduction seem to have the largest impact if you take individuals compared to other interventions that people have tried. So that's kind of the baseline. If you can improve that, that has the largest movement in terms of epigenetic clocks, in terms of outcome measures. That's kind of a baseline to set the stage. So if you can make a move there, that remains foundational, I think, to epigenetics. And, I think we still are awaiting more targeted therapies in terms of what could emerge from this area of research. There's an emerging repurposing effort going on clinically with FDA-approved trials, where people are going back and looking at things that seem to maybe impact aging. The one that's received the most attention has been metformin. The other one that's kind of at everyone's thought right now is also these GLP-1 receptor agonists for weight loss. So there's some thought that if we can repurpose those in people maybe that don't need weight loss or cardiometabolic benefits, that might slow down their aging rate potentially. But again, unless you have the randomized clinical trial data, it's still anecdotal or observational. So I would not make that recommendation at this stage. The best evidence currently is still I think those three that I've highlighted up top, which for those that study brain health, those are also a large part of what's impacting the social determinants of health and how that interplays with our brain. And then today, I just hopefully wanted to highlight briefly, this idea that was presented by prior presenters as well. These epigenetic clocks have kind of emerged recently as an informative biomarker of the gene by environment interaction. So it kind of gives us a way to distill down one measure of a lot of things occurring in the environment, and you get kind of a number that essentially can tell you, okay, generally speaking, this person may have a higher aging rate, which could associate with 50 other variables, but we now have kind of one biological measure that we're able to kind of capture that in a composite way to track things over time. So currently we're doing a lot of work to utilize this, both here in collaboration with Nano and others as well. I see David, nice to see you, in Medellin, to look to see whether this measure has impacts in terms of Alzheimer's disease and neurodegenerative disease, to really help us understand maybe progression or other aspects of brain health in that space. So I would consider this as an informative biomarker. It's not a recommendation, to eliminate all the gold standard kind of clinical phenotyping, but it's more informative, I think, in terms of thinking about how over time someone's genes aren't going to change, but what will change is how they're using their genes. And this is what kind of this aspect is trying to do. And then just to end today, I think the presentation was really to make the point that I think that the epigenetics and the environment is a new horizon in neuropsychiatry. We're only kind of scratching the surface in terms of moving this into, I think other areas of neuropsychiatry that we really want to start to understand. And I think the more data we have and the more groups working on this and more students and people getting trained in this space, I think will really help kind of guide the future direction in terms of this landscape. So just acknowledge, I think with the multiple partners, and, I really wanted to thank Nano for the invitation and everyone here that, I think is in person, and the invitation to come here. Being in Colombia is my, absolute favorite thing to do. So unfortunately, I got delayed 9 hours. But, I look forward to talking with everyone in person, and happy to share any ideas and future kind of projects. But we're quite excited in terms of what we've been able to accomplish thus far. But, I think the next 5, 10, 15 years, this space is only going to continue to grow and really extend kind of the work that's being done here and the incredible progress that's already been made. So thank you.
Thank you very much, Dr. Corley. Is there anyone with a question?
Hi. See? Hi, Mike.
Hi.
Different to DNA methylation, there are another epigenetic mark that tell us information about aging or about like the epigenetic clocks, different markers.
Yeah, so they have chromatin modifications. So these are proteins that are attached on DNA that they've used to show that that's been kind of a way to develop another clock. You can kind of build a clock from a lot of different technologies in terms of proteomics and other things. The reason we've kind of not strayed away from that, but kind of not included that in a lot of our analyses, they're very difficult to replicate across studies because it's a very sensitive kind of assay to measure a protein consistently. Where the DNA methylation data, we've applied that in a lot of different studies in other groups as well, where, I think they've even extracted DNA from mummies in Egypt and looked at methylation patterns in Egypt, so you really get a very, very consistent measure because it's biochemically a covalent modification, so it's very stable. So that's why we've kind of gravitated towards that. And then the other thing is cost as well. So the cost for kind of these tests is slowly coming down now with some testing companies. I'm a little bit hesitant to kind of, I think get into that space just because, these are companies that are selling this to people that are interested just to know their age biologically, and they're testing themselves, just I think it's a curiosity they have. But, whether that information is providing any sort of, additional guidance to their, without the other clinical data that's more important, I think is unclear at this point. But yeah, and the other thing is large data. So we have, I'd say at least 1,000 times more samples for every, one data set of the other kind of modality. So, but.
Mike, thank you so much, Mike, for the presentation and for increasing our, knowledge and awareness about the potentiality of this kind of methodology. It works? Okay. My question is about it. We are totally convinced of the power of biological clocks and epigenetic clocks. But I was wondering if there's another, epigenetic techniques that could have like a potential, options to be used as a biomarker in clinical settings. I was wondering if, I don't know, the EWAS approach or the genes approach in Asian, like in the future, if you see that kind of future, like an option for guidance settings.
Yeah, I think so. The one that I would look forward to getting more kind of data and information from is, so from a blood sample, we're just looking at the DNA that's in your immune cells. So we're just relying on the immune system telling us what's going on in the body, and it's fundamental medicine immunology. The immune system tells you something. So that's what we're capturing. The one that's emerging right now is in the plasma, which people are doing a lot of great biomarker space for Alzheimer's disease with p-tau. In the plasma of a blood sample, what we're also finding, which groups are starting to now look at, is cell-free DNA. So DNA coming from cells in the body that die, that gets into circulation in the blood. So for that, you can look at the epigenetic patterns of those. And it's a little bit different concept, but the concept is if a brain cell dies and that cell needs to be kind of cleared through the body, that DNA of that brain cell has a very specific methylation pattern. So we can see in the periphery what that pattern looks like. It's been very informative if you look into the oncology space. So some of the best cancer predictors are these cell-free assays, which also use epigenetics. But now people are starting to look at other disease states. And the brain is kind of emerging as one where we've done a little bit of pilot work in cerebral spinal fluid and its work. So in your cerebral spinal fluid, you don't have a lot of immune cells, but you have enough to get DNA. And it's kind of an understudied compartment from like immunology. People really focus on cerebral spinal fluid, the fluid. And unless they're immunologists, they usually just discard the cells or, they don't study those immune cells. But within that kind of CSF fluid as well, there's this cell-free DNA that people have been looking at recently. But it's a space I think that, it's wait to see kind of larger studies emerge. But at least the data I've seen has been pretty exciting in terms of another way to think about another epigenetic marker that can come from, not the cells themselves, but from other parts of the body without having to do like, a biopsy, essentially. It's kind of a cell-free biopsy, a liquid biopsy, they call it. So.
I just wanted to respond a little bit to Christian's question. So some people in my group have now started making clocks from RNA sequencing. But it turns out, I think cost is a big issue. It's been very, it's very expensive to get RNA sequenced.
Yeah.
And also we're trying to do a lab validation exercise because we also don't know if each lab is running RNA sequencing. Whereas DNA methylation is fairly standardized. I was at a meeting, a biomarker network meeting a couple weeks ago in Washington, D.C., and Illumina is creating a new chip for us that has more like aging-related sites because the chips were originally designed for cancer research, and so I think that's really interesting.
Yeah, I heard through the grapevine, because I'm in San Diego, so they're right down the street. There's larger, yeah, it's like a couple million, I think, CPGs it's going to assay. So, but.
And the other thing that we've been doing is work on DNA surrogates. So for instance, maybe one study has collected urine so they can measure lead exposure and they can get the CPG sites, and then we can check those CPG sites in our blood data that we have. But like, for instance, here, Nano, in your samples, you could check to see if there are CPG profiles related to people who've experienced violence and displacement. And there's a study in Northern Ireland that has a similar internal civil conflict called Troubles, and they also have epigenetic data. They also have proteomic data, which no one else has. That's also very expensive.
Yeah.
But so I think that Christian had a good question. There's lots of new things out there, but it's mostly like cost and also our ability to do comparisons across labs
cohorts,
and groups.
Yeah, great point.
Thank you very much for your presentation. I'm just wondering, most of those epigenetic clocks have been developed from data from European population or North American population. So would you expect it to be different in our mixed population, the patterns of CPG size methylated differently right here?
Yeah, so there's a lot of beautiful work showing kind of admixture has an impact. And depending on kind of the clock, I guess I would tell people I'm not a clock builder. I did it just kind of as a curiosity. We built it based on these endogenous retroviruses because I have this other expertise where we've been looking at brain infections. But in that space, every piece of DNA we have embedded within our DNA, it's referred to as dark matter. So these transposable elements that through evolution were inserted. So old viruses got into the germline and they've been shut off and silenced. The Alzheimer's space has done a lot of beautiful work with, they're called line 1 elements. So these elements start to turn on as people get older, and there are these jumping genes that move around. We just looked at the clocks that were there, and I'm like, there was a lot of biomarker interest, but mechanistically, none of the genes made any sense to anyone. So no one was tracking anything that made any sense. It wasn't like an aging gene. So we basically filtered all this data of like 12,000 people, and we only looked at these retrovirus elements. And we could build a beautiful clock that worked perfectly, just as good as every other clock. But it was because these elements were starting to turn on over time. So there's kind of this global, I think, change that happens with age. But depending on where it occurs, I think it's fundamental. And then, in terms of kind of thinking about the clocks people are developing currently, there's always going to be a new clock every week. It really matters what's the question you're asking, I think, in terms of using the clock. So we've typically been using the clocks for looking at interventions. So if the clock is miscalibrated, say here in Latin America, that miscalibration will still be responsive to intervention. So you're going to be off in terms of maybe you're underestimating someone's age, but you're still underestimating their age longitudinally. So I'm a big fan of people that are doing longitudinal studies. We're using an individual and comparing against himself. Not to say the cross-sectional studies haven't been important. It's just, I think that's a little bit of a different question in terms of trying to associate disease with aging processes versus looking at an individual over time and trying to track kind of trajectories of people. So I think just depending on your question, I would just kind of, gravitate towards the ones that are a little more used, which I think would be the mortality ones, or ones that are shown to be responsive. But there's definitely no harm in kind of, trying to improve the clocks with more data. And that was really kind of what we had hoped, I think with our effort, we're slowly kind of building more and more data. And hopefully, I think there's been a new initiative too, with a lot of people from different groups having access to try to combine the data in a way. But with that too, it needs a little bit of annotating in terms of going through the data in a way that makes it compatible. So, I think back to the other kind of point that was made, the clocks we were building on these new retro elements were using brand new chips. So we had essentially double the number of sites to train on. And it was more interesting in terms of that measure. We were only looking at 1% of the whole DNA code, which is 23 million CPGs. So we're just scratching the surface. But the fact we still get a signal and everyone else is seeing signals, to me, it's kind of remarkable because it's really kind of, there's something there. it's just a matter of, in terms of using it in a way that is for a question, I think, that you're interested in asking. But yeah, I think the more data we can generate and get groups to think about it, which right now, there's a lot of data there, but I think we're starting to see some of that change too with, if you're interested, there's 2 very nice papers that were just published. They've made these methylation language model equivalents. So you can query methylation patterns similar to how people are using ChatGPT. And it's essentially like epigenetic GPT. So it'll tell you, using all the data that's out there, what a reference methylation data point should look like, which is kind of an interesting idea. But with it too, its use case is limited to the data they had. So it essentially can fill in data that they had from other data sets, but the missing data, the imputation doesn't seem to work that well at this point. But yeah, I think it's only going to increase over time. And, I think people working on it too from other spaces, that's where the big breakthroughs happen. So, some of these people that are from, language model development, they maybe know nothing about epigenetics, but the data is a data format they can configure into that kind of format. It's really helpful for the field to think that way across discipline and stuff.
Queda tiempo para la última pregunta.
Thanks for your talk.
Thank you.
Has been described that the neuropsychiatric disorders produce epigenetic heritage. Thinking in biomarkers, the very early biomarkers based in epigenetics, do you think that epigenetic heritage could be interfere or could be give very early highlights about the susceptibility or predisposition to the future disease related with Alzheimer or other neuropsychiatric disorders and the young people?
Yeah, beautiful question. I agree completely. I think that's been a leading hypothesis in terms of looking at susceptibility and it being a biomarker of predicting risk of neuropsychiatric disease. We haven't done a lot of work in that space, but there's a lot of literature in terms of looking at bipolar disease, looking at different neuropsychiatric conditions and what happened in youth and looking at the profiles there and predicting out. The one difficult part, which has kind of been, I think, something we're trying to sift through, or the field is, which I did early in my PhD, there's a very clear difference between what's in the brain tissue and what's in the blood. So unless the neuropsychiatric disease, which I think a large part of them do have a neuroinflammation or immune component to them, and that's now seen, I think, being even more appreciated in the Alzheimer's space of immunology having a large impact on brain health in, Alzheimer's disease. Similarly, I think with the neuropsychiatric field, if it has a component there that you have an altered immune system, those markers seem to be very good in terms of predicting kind of neuropsychiatric disease risk. Where it doesn't perform as well is sometimes in neuropsychiatric disease where maybe the change needs to occur in the brain itself in early development. And for us, we saw a lot of that actually looking at autism spectrum disorder, where a lot of these epigenetic changes we were observing not in the blood, but actually in the tissue of brain tissue. And really what it fundamentally came down to was it was the brain stem cells where the signal was, and that brain overgrowth that occurred in autism spectrum disorder was related to that signal being there. So when their age came where they needed to start to, have brain growth, they kind of overshot. So their brain grew bigger than it should have been. And the epigenetic profile was kind of predicting that kind of growth response. And really what it was is the stem cells maintained being a stem cell. So they just kept dividing. And then on the other side, the signal for pruning, where you needed to prune down the connections, that's where we saw some signals too. And others have kind of observed that kind of dichotomy. But yeah, I think it depends on the neuropsychiatric disease in terms of what the potential biological piece, but I do believe the immune system is a large piece of a lot of it. So I would think that, if you have a lot of diseases together and enough data, you could potentially look at general risk of, neuropsychiatric disease in total by doing it that way. And, I think that's what another important thing of this meeting is, kind of bringing different experts across different domains. Cause, I'm coming at it kind of from trying to think about aging and But if you think about that process, usually there's very specific experts in cardiovascular disease, the GI tract, different organ systems, but neuropsychiatric disease as well. It's kind of, if you can break down some of those silos, you see things are somewhat common, and then there's very specific, unique pieces. And that's kind of, I think, approach that, it's really great to see kind of, different experts being able to kind of share data to say, hey, I see an immune signal here, or, other components, which I think is great to think about. Yeah. Yeah. Thank you.
Thank you again, Dr. Collier. I think that presentation worth the wait. Before we leave, we have a special thanks to give. Please, thanks to our sponsors. Thanks, they made this possible. Thanks to Dr. Santamaria. I think he's the main reason that a lot of us are here today. Thanks also to all the presenters, the people who presented today. And a special thanks to my co-host, who is today, it's her birthday. So please, a special applause for her. We hope to see you tomorrow at 8 a.m. at this same auditorium. We will begin at 8 a.m. as I said, and thanks a lot for remaining with us.